Morning, sir.
Good morning. Very good morning. We'll wait for 1 or 2 more minutes and then start.
Good morning.
Good morning. Good morning, sir. Good morning, everyone.
Everyone, yes, good morning. We'll start at 9:32. 2 minutes grace.
So shall we start?
Yeah.
Okay. Good morning, everybody. And I would like to start with thanking our Honorable Vice Chancellor, because he is the one who initiated this. Sorry, there are so many admins. Okay, who is the one who initiated this program first time in the University of Hyderabad at a grand scale, and he is the one who is behind everything, except we did a little administrative work or whatever it is. Okay, I would like to welcome Professor B.J. Rao Garu, Vice Chancellor, University of Hyderabad, for giving introductory remarks. Thank you, sir.
Namaste. Good morning to everybody here from university as well as from outside the university. We are celebrating the Nobel Awards of 2021. It's a celebration of 13 awardees, 13 in total. And celebrating these awardees would be to discuss what they have done. That's the best way we celebrate. There are about 10 speakers in this morning session describing in nutshell why these people were given awards. And we have 2 senior faculty members chairing the sessions, one for sciences and another for non-sciences. And we have 2 young coordinators. I want to thank all of you. I want to thank all the participants for making this an eventful and an enriching process. Alfred Nobel Award came from dynamite chemistry. In some sense, chemistry is the reason why all these awards are given. And this is about 120 years of legacy that we have been enjoying these awards. About 943 individuals received these awards so far, including this year's awardees. And out of these, 9 awardees from Indian origin members. So you can see that we are seriously underrepresented in this journey. And the awards are basically given to those people who have made significant contributions for the benefit of mankind. So I think it will be a fantastic tribute. We discuss their contribution and we cherish their contributions, and hopefully in the years to come, Indian contribution would significantly increase. Thank you very much. Namaste. Good morning to everybody again. Over to you, Bindu.
Thank you, sir. Now I would like to welcome Professor K.C. Kumaraswamy from School of Chemistry to take over this chair and for this first session. Thank you, sir. Please.
All right. Okay, dear friends, welcome to the symposium again. Right, in the first session, we have topics of physiology or medicine, physics, and then chemistry. Time allotted to the first 2 topics are 25 minutes for the presentation and 5 minutes for the question and answers if there are any. And then for the third one, it is 38 plus 7 minutes for the last topic, that is chemistry. I hope that the speakers will maintain their time. I also hope that this symposium will encourage all of us to do some useful work to your society, and this was the primary aim of Nobel. So I hope that we will be able to do something uh, in our lives too. Now, as far as the first topic is concerned, that is physiology or medicine, there are three speakers in this one. It may be a difficult thing for them to organize. I hope that they will be able to organize well. The speakers are Dr. Madhubabu, Dr. Prasad, and Dr. Joby Joseph. I will give a brief, very brief, what can I say, CV with respect to these three speakers. Okay, Dr. Madhubabu is assistant professor in the Department of Biotechnology and Bioinformatics in our university. He did his PhD in molecular biology from Max Planck Research School of Planck Institute for Biophysical Chemistry in Göttingen, Germany. His research interests include neurodegenerative diseases and behavioral neurosciences. He has a few awards, okay, to his credit. Dad, namaste. And he also was having an internship at the WHO, World Health Organization. Now, coming to the next speaker, Dr. Prasad, he's a professor in Department of Biology at the University of Hyderabad, is our university. He did his postdoctoral research in Rutgers University, USA, and he was also a Pickover Research Fellow at MIT. His research interests include molecular neurosciences, molecular mechanisms of membrane trafficking, and how organelle protein quality control is regulated in neuronal health and disease. He has funding from ICMR as well as CERN. The third speaker for this talk is Dr. Joby Joseph. He is an associate professor in Center for Neural and Cognitive Sciences in our university. He had his PhD in electrical communication system from IISc Bangalore. He had his PhD assistant at NIH website, USA, on neuronal information processing. He was also a Ramanujan Fellow in the year 2011. His research interests include the topics like how is information processed in neuronal systems, algorithms, and computational principles that come into play, leading to perception and behavior. He was also a DID. He also had a DID fellowship in Germany at the University of Kaiserslautern. He has more than 16 publications, with one of them being in Nature Neurosciences. Now, I will leave the podium to the speakers to start their talk. Your time starts now.
I request speakers to be close to the speaker and then be little louder, because there is a lot of voice issues, internet issues.
Yeah.
Thank you.
So, good morning, all of you. And thank you, Professor Kumaraswamy, for introducing all of us. And so, I also thank organizers for giving us wonderful opportunity to share this wonderful insights that were made by the Nobel laureates of 2021 in physiology and medicine. I'm going to share my window of presentation. Okay. Okay. So is it visible and my voice is clear?
Yes, yes.
Yes, very much. Go ahead, please.
Yeah. So good morning, all of you. In the next 30 minutes, we are going to discuss about the contributions of Dr. David Julius and Dr. Ardem Patapoutian in the realm of neuroscience, and that resulted in finally getting them a Nobel Prize in Physiology and Medicine in 2021. Dr. David Julius is a professor at the University of California, San Francisco. Dr. Ardem Patapoutian is a is a scientist at a Scripps Research Institute in California. So their studies actually unraveled mechanisms of thermo and mechanosensations. That means how's the sensations like heat and cold, and also the touch and pressure are measured or sensed by our body. We know we use different sensory organs to sense the environment around us. For example, we use different sensory organs for vision, auditory functions, and for gustatory and olfactory functions, and also for somatosensation. So there are different types of stimuli that stimulate these sensory organs. For example, vision is stimulated by photons. The stimulant for auditory sensation is mechanical, and the smell and gustatory sensation are stimulated by chemicals at the fundamental level. And and the sense of touch, the mechanical and thermal sensations the touch is caused by mechanical and thermal sensation. And the mechanical and thermal sensations can be combinedly called as somatic sensory system. And the contributions made by David Julian and Adam Pataposhian were into understanding the molecular nature and of these receptors of these mechanical and thermal stimulations. Okay. So in our body, we have a skin and other visceral organs that sense the touch mechanical stimulus, as well as the thermal stimulus like heat and cold. And the sensations from the skin and visceral organs, they travel through primary afferent neurons. The axons of these, you can see this track, and they all are clubbed, and they all go together in the same pathway, and they reach a region called dorsal root ganglion, and from there they go to higher parts of brain. So initially the signal will travel to cerebral, vertebral column. From there, from spine, it is then reached to the brain. For all these sensations from different sources, the stimuli from different sources travel, and then the brain then combines all these stimulus and generates perceptions. On the right, you are seeing a diagram, hand diagram by another great Nobel laureate who got insights into the structure of nervous system, that is Raman Kahal. So the sensations from here are traveling to the spinal cord, from there to higher parts of the brain to create perceptions about our environment. To sense this, so for somatosensation, skin has a lot of receptors. You can see here the ultrastructure of the skin on the slide. On the top portion, epidermis, and the next portion is a dermis. It is all innervated by different types of neurons and the receptors, like some of different types. All these and also the receptors that are present in visceral organs, in our heart, lungs, and liver, and all these visceral organs also sense mechanical stimulus as well as thermal stimuli, and they send the information to the brain. So generally, when a stimulus is given to the cells, the neurons carry this information. When a stimulus reaches the cell, the stimulus is converted into an action potential, the electric current, and this electric current travels along the long structures of the neurons called axons, and it reaches the terminals, and the signal is again transferred to the next neuron. So basically, the stimulus is converted to an electrical, the stimulus is converted to electrical action potential, and that travels across and then propagated. Then it is generated and propagated. It is generated and propagates and reaches the brain. So when the stimulus comes, what actually happens at the cellular level on the neuron is that there are ion channels, and as a result of stimulus, these ion channels open. When the ions channels open, they generate a flow of ions, and that causes the current. That is what we call as action potential, and it propagates to the cell. There are so many different types of ion channels. So you can see here different colors. So there are sodium channels, potassium channels, calcium channels, chloride channels, so many different ion channels that actually translate that chemical signal into electrical signal. So we can measure these signals by putting a micro electrode onto the cells. And we have a glass pipette with an electrode. We can put it, we can clamp the cell to it, and study the electrical properties of the whole cell or the single ion channel. And this technique is called patch clamp. And the discovery of this technique itself got Nobel Prize to Erwin Neher and Bert Sakman. So you can see when ions flow through this ion channel here, when it opens, you can see the generation of action potentials. And you can see the flow of current. When there is no current, you will have baseline here, and when there is a flow of ions, the generation of action potential, you can see that the channels open and the current flows. So how these David and Ardem went on to find the molecular sensors of temperature and pressure is the focus of our discussion today. And I'm just going to tell you what is the strategy they have adapted, and brief introduction to it, and I will introduce you the strategy first. So usually there are two assumptions for their strategy. First is that different neurons have different molecular fingerprints depending on their function. Okay, a specific sensory function would then encode as corresponding molecular sensors in the neurons that detects that particular stimulus. So the idea is to identify these specific molecular sensors of temperature and pressure. And to identify these specific molecular sensors, we need to first know the neurons that respond to the stimulus. So that we all know what we already know is we know the neurons that respond to the stimulus, but we don't know what actually it triggers that stimulus, what actually converts this stimulus into electrical conduction or generates action potential across the membrane. So to tell you about that, I have to tell you that all the genes in our body are made up of DNA, and this information is converted to RNA, and from RNA, the proteins are made. And if we find out any of these molecules carrying out this specific function, we can know the other one easily, because the sequence from protein sequence can be used to decode the sequence of DNA, and also from DNA we can also learn about the sequence of proteins. Usually, scientists use two different ways to understand, to find new genes or to understand the function of genes. So in the lingua franca of genetics, we call them as gain of function and loss of function. The gain of function approach, these are two approaches. In the gain of function approach, we have target genes, and we isolate these target genes, or we create a mutation, or we might be interested in studying a mutation, how that causes some phenotype. And we take out this gene, isolate this target gene, and put it in a vector and make a recombinant DNA. You can see here the gene is taken out. Then we introduce them into a different cell and check check their phenotype, like what is what is a new function that is attained by a cell, or what happens to the animal in which this new gene is added. Okay. So for example, if you introduce a gene of obesity, or if a mutation causes obesity, you would see a gain of function, like the animals will become obese, like in this picture, the white mouse. Uh, Another approach I said is a loss of function approach, in which we have either cells or animal. In this, we take out, we silence the genes by using different methods like RNAi, or gene knockout methods like CRISPR. After taking out the function of gene, or knocking out the gene, we'll check what is the effect it has on a phenotype. Okay. For example, if you knock out a gene that is involved in short-term memory, it will the phenotype would be that animal will forget the things easily. We can design assays and study how it works. Okay, so so basically, these gain of function approaches and loss of function approaches were used by David and Arden to find out the thermoreceptors and the mechanoreceptors. So how did they go about it? So we know the sensory neurons that carry the signal. They are called dorsal root ganglia. So these dorsal root ganglia should have that information that encodes these thermoreceptors or mechanoreceptors. And for that, what they did was they know they used a compound called capsaicin. And capsaicin is present in hot chili peppers. I mean, when we eat chilies, we get that burning sensation. That pungency is caused by a chemical called capsaicin. And we also know the cold stimulants. When we, in some oils and in some lotions, we have this menthol that gives us a sense of cold. So, and they use these stimulants, and we know that these cells will respond to that stimulus. And to identify the molecular players in it, they have initially isolated the RNA, the molecular fingerprint of these cells. And then cDNA was made, and this DNA was transfected or was used to transfect the cells that would be otherwise don't respond to these chemicals. For example, if you have a cell line that doesn't respond to capsaicin and menthol, the cDNA was used to transfect these cells, and gain of function was studied, because the gain of function would be if a correct DNA is there in the cell that encodes a molecular sensor, molecular sensor, then it would respond to capsaicin. So they then checked, and the presence of this molecular sensor was also detected by fluorescence. Okay, because there was a calcium sensor and capsaicin added, so the calcium sensor would fluoresce whenever there is a calcium inflow. So in that way, they could screen for the thousands of DNAs to find out the sensor, molecular sensor for capsaicin and also the menthol. And in this process, they found a receptors called TRP family of receptors, TRPV1 specifically, and TRP-mite. TRPV1 senses the hot stimulus from capsaicin. The same, they later found out that the same molecular sensor that responds to capsaicin also responds to another stimulus that is temperature. So when you heat up, these channels also respond. So they are also they respond to cold, and the receptor that responds to capsaicin also responds to hot temperature, heat, and also the receptors that respond to menthol also respond to cold temperatures. Okay, and then these were coded and these were characterized further. And for loss of function studies, the loss of function screens were used for studying mechanoreceptors. Initially, the cells that that respond to mechanic stimuli were identified, and then the several candidate genes were chosen. Basically, they focused on ion channels because something that responds to mechanic stimulus should be present on the cell. And one by one, they knocked out the candidate gene and checked the effect. So if the candidate gene is required for translating mechanical stimulus, then there won't be any stimulus. By using this technique, they identified two receptors called piezo receptors that respond to mechanical stimulus. So now I would hand over the discussion to my colleague, Joby. He would talk about temperature reception in detail. Followed by that, Prasad would talk about mechanical stimulation in detail.
Please make sure that your time is maintained. You have to maintain the time.
Yeah. So as Madhu told, we know the sensory fibers which are responding to heat will also respond to capsaicin. And so this is where Julius group starts the work. And we know those neurons are actually located in the dorsal root ganglion. So which means, as Madhu said, if you want to extract the RNA which would be corresponding to the protein which would detect this sense, we need to isolate the RNA and possible proteins corresponding to that from this particular class of cells. So the procedure is as he described. We take out this DRG, dorsal root ganglion, extract the RNA, all the RNAs which may be expressed in that, mRNAs, from which we synthesize the corresponding DNA complement to it. And so the idea is that if this DNA, one of them is corresponding to the gene that responds to heat, which is also responding to capsaicin, when we put capsaicin in those cells which expresses the protein corresponding to this cDNA, it should light up because it has a calcium sensor which lights up when this capsaicin activates the receptor. So, but there is a huge number of candidate genes which are present in this DRG neurons. So we have to selectively find out one out of the 16,000 clones. So you don't do 16,000 experiment, you split this into subgroups which are called pools, and then transfer them into the cells. And if one of the pools shows up some lighting, meaning so the candidate gene is in that pool, then you split that pool further. And so on iteratively, if you split, you will come up with that one gene which is giving rise to fluorescence in all the cells transfected. So that's the way you go down from 16,000 possibilities to that one gene which might be responsible for capsaicin as or temperature. Now, once you have identified that, we want to see whether that's indeed showing the properties which are exhibited by the receptor. So we express that protein in a particular kind of cell which doesn't have many other channels. And then when we apply capsaicin, they should respond by shown by these currents which are measured from the cell, as well as other agonists which may cause the cell to respond. Not only that, if you actually put heat on that cell, also it should respond. So the same cell now having only this particular protein which you have isolated is now responding to capsaicin, heat. And also if you block the things which usually prevents us from sensing heat of capsaicin, which is ruthenium red in this case, you should be preventing the currents. Right? Not only that, now we don't know whether this protein is there, but whether it is a single channel on this. So to know whether it's a single channel which causes opening and then causes these currents, we actually take a small piece of the membrane which may have that single channel and then see whether application of capsaicin will produce small currents, which means there is no cell, but only this protein, and that can cause currents to come in with the binding of the capsaicin or temperature. So now if you have the candidate gene which is established to be responding to capsaicin, and we feel chili hot when capsaicin is from chilies, so that means that if you apply different kinds of chilies onto this cell which expresses this receptor, then the proportionality of the responses of the currents should somewhat correspond to our proportionality of our sensation of heat to these different kinds of chilies. So they took these different chilies which are rated to have different kinds of heat sensation for us, which is actually read as, where did it go? Okay, so that rating is called Scoville rating. So these chilies are different Scoville ratings, and you can see that roughly the Scoville rating is corresponding to the current induced in those channels, in those cells expressing this channel, which means that somewhat this channel is responsible for a perception of heat, but it is not perfect also, as you can see, which means there could be other mechanisms which might be also contributing to it. Now the question is, if we have identified this protein, then this protein should be present in the dorsal root ganglion. So those proteins as expression is checked, looking at the ganglions which may be having those cells, which are dorsal root ganglion and the trigeminal ganglion. And you see this red color indicating indeed the presence of those RNAs. And then the question is, we know that this particular cell will exhibit both response to heat as well as when we get injured. For example, when there is an inflammation which causes pH levels to change, then these compound sensation of heat and sensation to capsaicin. So if you apply heat and then change the pH, you can see that this cell respond, indicating the same sensor is responsible for sensing the pH change under heat, as well as this under this lower pH, which is acidic, capsaicin has stronger response, indicating that inflammation can sort of be explained by the presence of these channels. And heat plus the capsaicin will have a stronger effect, even in an habituated cell. This is the explanation for inflammation. So if inflammation takes place, then protonation, when basically acid is increased, that increases the sensitivity of the channel, which means after that, if you give the same amount of heat, you will feel higher sensation because higher current going in through these channels. And that is a work done by Julius' lab. At that point, now we have this particular protein identified, and which is homologous to many other proteins which are called TRP channels. So that heat one was TRP V1. And so using the similarities of the protein structures or sequences present in there, Ardem Petaproteins lab then identified another channel which is present in the dorsal root ganglion, which was responding into a particular, the mint or the menthol, and that they call TRPM8. And which means that if you similarly, if you isolate that protein and express it in some cells, these cells should respond to both the menthol as well as decrease in heat. So here is showing the decrease in heat or cold actually causing the cells to respond, indicating that this channel is sensing a decrease in temperature. Same way, it's the same channel, because it is supposed to be having hallmark of it is to also be activated by menthol. You can see that menthol also causes activation of the channel, expressing this TRPM8. And this menthol activation is temperature sensitive, meaning that at normal temperature, the menthol may cause us to feel cold, but it may not do that at higher temperatures. Now, do the rats actually or mice actually use this sensor? So we can create knockout mice, as Madhubabu said, that you can remove this particular gene TRPM8 from the mice. And in those mice, if you look at the nerves which leave the DRG into the brain, you see that in the normal mice, you get a lot of activity when the temperature is decreased. But in the case of mice lacking the TRPM8 channel, you find that hardly any activity is present when the temperature is decreased. Now, actually, whether this really reflects in the behavior of the mice can be tested in this kind of behavioral assay, where there are two halves of this cage, where the temperature can be controlled, or of the temperature of the floor can be controlled. And if you keep 30 degrees on both sides, then you see that the mice will be present on either side roughly 50% of the time. Now, in normal mice, if you decrease the temperature from 30 on one half of the cage, then you will find that it prefers to stay in the 30 degree for which it is comfortable, which is true that it is in the normal mice. As the cold is increased, will prefer to stay in the 30 degree window. But if in the TRPM8 minus, which is lacking the TRPM8 channel, you find that even when the floor is made cold on this half, the mice does not sense the cold, and so it actually goes to both sides equally. Right? And we have to decrease the cold below 10 degree, which is actually a sort of noxious cold, for the mice to actually sense the difference in temperature and stay more time in the 30 degree half. Now, this means that there are a family of TRP channels, and different channels actually are activated to different extent with different temperatures. So you might actually think that, for example, if you take 20 degree, TRPM is open and then other channels may be closed. Or if 40 degree, this set of channels will be open. But this is not necessary. So which means we may think that this activation is what causes us to. Is it over? So I will hand over the speaker to Prasad.
Oh, hi everyone. I think without much further ado, I think let me talk about the contributions made by Adam Pataputian to understand how the mechanic sensation is being perceived by the body. So before going to that, let me give a very quick introduction about what the touch is all about, right? So as Madhu Babu was pointing out, the touch is like, another mechanosensation property of the body. But unlike other senses, the touch is actually, perceived by the mechanical stimuli rather than chemical stimuli, right? And this touch is like not something new that we hear. Like, we often experience this touch quite often in our daily life. Like, if you want to feel the texture of something, you use the tactile discrimination. And if you have some emotional breakdown, you definitely rely on affective touch. And besides these two, there is another interesting aspect of the body which is called proprioception. In this proprioception, you can tell where your limbs are with respect to your body without even looking at. For example, I can move my hand here and there, and I can definitely tell where my limb is with respect to my body without even looking at. So this proprioception is very, very important, and we use this quite often to do many complex tasks in our daily life. For example, whenever you are climbing stairs, you don't even look at your legs where they are, and you can definitely know how high you have to lift your limbs up so that you can climb the stairs. Okay, so the touch is like a very, very important aspect of our body, and it is being perceived very well according to the external world, right? So looking at the biology perspective of the touch, the touch is being sensed by sensory neurons, as Madhu Babu and Joby was pointing out. Whenever you touch something, then these signals were taken by the sensory neurons, and these signals will be perceived, decoded by the brain, and send the perception signal back to the place where it was sensing. In the case of tactile discrimination, this is what happens. In the case of proprioception, the tension in the muscles will be read by the sensory neurons, and depending upon how much tension they are being elicited, you can tell where your limbs are with respect to your body. Okay. So far, this part of the biology is very well known that whenever you have mechanical stimuli, your sensory neurons take the signal to the brain, which decodes and converts that into a perception. But what is not known here is before Adam's work is that here basically your mechanical stimuli, you are converting that mechanical stimuli into the language that brain can easily understand, which is electrical stimuli. But what is not known is who actually transduce these mechanical stimuli into electrical stimuli. That is the main crux of Adam Pataputian's work, where he was really interested to find out which protein is actually transducing this mechanical stimuli into the electrical stimuli. So to understand, to identify that protein involved in that, so Pataputian's group actually took a reductionist approach. By this, the first identified the cells which are responding to the mechanosensation. So what they do is they'll just take the cells in a dish and they just poke the cell with some kind of a glass probe. Okay. On the other hand, you will just patch electrode and you read the current. If they are responding to the mechanosensation, then they open up the channels and they will just have the inflow of ions, and then you will see the current. So they just do that. So you'll just poke one side and just read the current from the cells if they are responding to the mechanosensation. Okay. So by this approach, they identified at least 72 candidate genes, and then they started knocking down one by one, and they identified one such protein family called FAM28A, which was not known earlier, and they named it as Piaggio-1, meaning pressure in the Greek, since it is sensing the pressure or the mechanical sensation. So whenever they knock out whenever they knock out the Piaggios, as you can see here. So this is the cells which has a Piaggio protein. They'll have a very good current. Whenever you knock down Piaggios from the cell, you can see that these cells are no longer responding to the mechanosensation. And then later they also find another homologue of the Piaggio-1, which is a Piaggio-2.
And piezo 2 is specifically expressed in sensory neurons, whereas the piezo 1 expressed both in sensory neurons and also some visceral organs as well. So after identifying the channel that responds to the mechanosensation, then they went on to devise the structure of the piezo channel, and they found that, in fact, they were quite surprised that this structure itself is primed to sense the mechanical force inside the membrane, right? So if you look at this structure, so you can clearly see that it has a membrane ion conducting channel in the middle, and then they have three more propeller blade-like things that are extending out. So whenever you have a mechanical force in the membrane, these propeller blades flatten out, and then there will be flow of ions into the cell, and which contributes to the mechanical activated currents. Okay, so this is a very rare situation where identifying the structure itself give a lot of insights into the function of the channel itself. And then to provide the direct evidence that this piezo 2 actually involved in the mechanosensation, the Petaputians group actually knocked out piezo both in skin mechanosensitive cells and also the sensory neurons, and found that these the piezo knockout animals, so they don't really display much currents when they were given mechanical or light sensitive touch. Okay. And this is like very conserved, as removing a piezo from Drosophila also display similar phenotype, where whenever you take out piezo, you don't see any currents, mechanical activated currents, indicating that piezos are actually the mechanosensitive channels that are there inside the body. And since proprioception is such an important aspect of mechanosensation, they went on to identify whether piezos are also involved in the proprioception. And they knock out the piezos from the mice, and they found that whenever you just lift the wild type mice, so it extends its limbs towards the down. But if it has a problem with the proprioception, then it won't display that phenotype, the very phenotype. And these mice are doing pretty bad here. As you can see, this is a wild type mice which is healthy and running around very nicely. But when you take out piezo, it has problems running around, as it cannot really elicit the proprioception property of the body. And the Petaputians groups also went on to identify the mutations in the patients also. These patients have very severe problems discriminating the touch, and also they have very problems eliciting the proprioception. So for example, if you see this human subject here, if she is blindfolded, she cannot walk in the straight line because she don't know where her limbs are with respect to her body. And similar is the case when you just blindfold her and ask her to find the bottle just in front of her, she will have very tough time finding it again and again, which is mainly contributed by proprioception property of your body. So subjects which have the piezo 2 mutations or loss of function mutations, they have serious problems with the mechanosensation and proprioception as well. Okay, so far they pretty well established what is how the piezos are involved in sensing external touch and mechanosensation. But interestingly, in the body, there are so many tissues which also respond to the mechanosensation. For example, when you eat a tummy full of food, your stomach expands. When you have to urinate, your urinary bladder also expands, and it generates a lot of mechano force on the cells surrounding that and the sensory neurons as well. So in order to see whether piezos are also involved in these sensing the mechanosensation inside the body, which they call it as interoception, they went on to do so many experiments by removing piezos in all visceral organs, and indeed found that whenever you mess up with the piezo's function, so they actually have serious implications in interoception property of the body, where you have respiratory distress if you remove piezo in the lung, or you cannot really control your blood pressure if you remove that in arteries or sensory neurons innervating arteries. Okay, so given the importance of this piezos in this, it is not surprising that either gain of function mutations or the loss of function mutations of piezos is actually associated with so many diseases as such. Okay, I think I will maybe summarize here. So to summarize, I think the discovery of these trips and piezos actually really provided us insights on how we actually sense and perceive the heat, cold, and mechanical forces. and adapt accordingly to to the world that we live in. And we are like hopeful that this research will definitely open up the floodgates for more physiological and pathological functions of these receptors, and possibly give some hints on developing the novel treatments on treatment regimens for conditions especially like chronic pain. So on the side note, I think this prize is a very good example of how one can do wonders by coupling, curiosity-driven basic research with the perseverance and motivation, so as to translate the information from the cellular level to the human genetics and medicine. And thank you, and we'd be happy to take any questions. And just sorry for not sticking up this time much, but thank you so much.
All right. Now the forum is open for questions and answers or comments. All right. Okay. 2 or 3 questions or comments. Time is up a little bit. Anyone? Yes, Tushar.
Thanks for the nice presentation. My question is very simple. Is there anything done on the time scale of these responses? For example, temperature or mechanical responses. I mean, how long it takes to recognize? Because that correlation between time and these responses may lead to identify one person to another person's response.
The time scale of total response can actually depend on a large number of factors, but we can actually characterize the time scale of the response which this particular class of neurons take after application or encountering the stimulus. And they are in order of milliseconds. But the actual response which the person can, one is about feeling the sensation, but other is about the report of the perception of the sensation. So the report can actually depend on other factors, but we can definitely find out the time taken for it to reach the brain, as well as time taken from the touch up to the the neurons actually getting activated. So all this can be characterized. There might be personal differences, individual differences, but by and large, I think up to the, from what we understand, unless there are some very strange phenotypes, they will be similar up to the brain. That's what I would think. But after the brain, what happens can depend a lot on the experience of the individual.
Yeah, and also it depends upon how strong the stimuli is also.
And also the type of neurons. There are different. Some neurons are specialized for translating, amplitude of the pressure. Some neurons are there specifically for measuring the vibration frequencies of the touch. So yeah, all these factors, time and specificity, and the type of neurons, all these affect the response and the speed of the.
All right, Vivek.
Yeah, Dr. Prasad just talked about the piezo receptors. What I would like to know, because the cell membranes in itself, they are not very static structures. They are fluidic or dynamic in nature. So how piezo protein distinguish those dynamic behavior of membranes against the mechanic perturbations?
Yeah, I think Vivek, that's a great question. So yeah, it's definitely the fluidic environment of the membrane definitely plays a role. But so far, there are not many reports there. But I'm pretty sure, the lipid composition and stuff like that probably will have much prominent effects on how it really senses the kind of tension, the force, or stuff like that. But definitely the fluidity will have definitely some role. I think probably more studies will follow.
All right. Yogesh Sharma, this is the last question. Okay. Others can contact the speakers by email or something like that. Yogesh Sharma.
Yes, sir. Am I audible, sir?
Yes, please.
Yeah. Sir, this is a very layman question. Does piezo also play a role in something what we all commonly known as a muscle injury or a pain which I may get in my day-to-day life, which has no outside or something piezo messing up a kind of phenomena? So does it relate with our daily life experience or completely different?
Yeah. So yeah, I think it's see the pain and mechanosensation. I think they do like go in hand. At least in the case of mechanical allodynia, where if you have a skin burn, just a simple touch is also painful there, right? So there actually the Piaggios, the role of Piaggios were actually shown there actually. So if you yeah, bradykinin also like an inflammation induced touch, stuff like that. If you have some kind of sunburn or like anything, just a simple pain also becomes painful. And the role of Piaggio was definitely it has a bigger role there actually. It was already shown.
So, like any further questions, you can ask the speakers by email or something like that. Now we'll have to go to the next topic, that is physics. Okay.
Thank you.
The speaker is Dr. Ashok Karumuri. He is professor in Center for Earth, Ocean and Atmospheric Sciences, University of Hyderabad. Okay, he's a PhD from Andhra University. His past positions are really interesting. Okay, so he was a senior scientist at the Indian Institute of Tropical Meteorology, and he was also senior scientist at APC Korea, researcher at Jamstec, and he was a postdoctoral researcher at Jamstec again. His research interests include Earth system modeling, climate change, modeling and applications in policy. Essentially, it's all related to climate. His honors include this. He is a fellow of the National Academy of Sciences, Allahabad. Okay, he has a large number of publications with a huge number of citations as per Scopus as well as Google Scholar. Now Google Scholar. I will leave the podium to Dr. Ashok to have his presentation.
Thank you, Professor Kumaraswamy.
Yeah, please maintain the time.
Yeah, I would. I'm covering at least I think works of two people, but I'll be mainly covering Professor Manabe's work. Is my presentation visible?
It is visible.
Yes, sir.
Yes, sir.
It's full screen now.
Yes.
Some fonts are of small size, but they are visible.
Yeah, yes.
Okay, fine. Yes. Good morning, everyone. This time, the award for the Physics Nobel was shared by three people, and very happily, Professor Sukiro Manabe and Professor Hasselman have shared half of it for the contributions to the climate science. As a climate modeler, and the whole climate community has been aware since last 3-4 decades that this is one of the most complex mathematical and physical sciences consuming one of the biggest uses of the supercomputing. But a lot of progress has happened, and Manabe's own words, from being from art, what you call as more of an art weather prediction. It was like that a century back to the pure sciences, in the sense, quantification in terms of that. So it took more than 1 century. And in the process, climate change research has been one of the major things. So I think let me some of the slides are. Let me just show you a couple. Slides are missing here for some reason. Anyway, doesn't matter. As I said, Manabe and Hasselman have shared the prize for the climate change, the contribution. So both contributions, Manabe's contribution essentially stemmed out from his contribution to the quantification of the effects of the greenhouse gases and how the moisture plays a role in recycling of the water vapor and, from the surface to the atmosphere, and most importantly, the development of the climate models. Okay. So now, how did it all happen? Further, all of you know that the climate change, the recent AR6 report, the IPCC report has come, and thousands of scientists take part in this one. This report came last month, and so you can see that within last two millennia, you see the sudden spurt in the temperature in the last 3 to 4 decades. This is unprecedented in the last 2000 years. And it's known. So we have unequivocally proven that it's because of the emissions of the greenhouse gases and all. Okay, so this picture from IPCC report also tells you that in future, depending on your CO2 emissions, the range of temperatures that you may expect. Of course, there is a certain amount of uncertainty, but this is based on about a few tens, if not hundreds of models, including for the first time, the Indian climate model has also contributed. Yeah, this is the slide I was looking for. So Manabe and Hasselmann for the physical modeling of Earth's climate, quantifying variability and reliably predicting global warming. And Manabe demonstrated how the increased levels of CO2 in the atmosphere lead to increased temperatures at the surface of the Earth. So he led the development of physical models in 1960s and was the first person to explore the interaction between the radiation balance and vertical transport of air masses, basically water vapor. So then, of course, current generation climate models. I'm not going through this one, but it's since the beginning of the 20th century, a lot of developments have happened as a as taking weather as a mathematical initial value problem. Then World War II fortuitously helped a lot, and new sciences have come. People started trying to give a prediction in 1922, but Richardson failed spectacularly, but that helped a lot to improve our knowledge and technology. And new dynamics, new simplifications help people to give weather forecasts in mid-latitude in 1950s, but that was more of a, giving a cyclonicity, etc., what we call it as. And now later on, model development has taken further observations and data assimilation. A lot of technological concepts and understanding of the chaos theory helped us a lot, and massive increase in the computing power. Some of the slides are from my former colleague Krishnan. And science of climate change, meanwhile, also started taking place since early 19th century, starting from Joseph Fourier. And the Tyndall's greenhouse gas, what we call as greenhouse gas effect, the atmosphere emits the entrance of the solar radiation, but it checks the radiation from the Earth's radiation. So there is a tendency to accumulate heat the surface of the planet because of the greenhouse gases. And then, Arrhenius quantification of response of temperature to the CO2, and Calendar's intervention that humans play a role, and Keeling's modern day observations with Mauna Loa that has been the standard for measurement of atmospheric composition. And Krishnan has put these two papers of Manabe and Strickler, Manabe and Weatherald, even IPCC quotes them. But there is one more paper, Manabe's convection paper. I also think it's less a fundamental path-breaking work. I will show some result from that. And before I go further, most of you know that when you take the rotation of the Earth, equations of motion, your Newton's familiar F is equal to ma is much more challenging because you have what is called as a Coriolis term, and there are a lot of nonlinear terms. With so many terms, you can't solve these equations. That's a story for some other talk. But importantly, if you see the forcing right term, you see, for example, this horizontal velocities and your moisture equation, temperature forecast, you see this red color forcings. Of course, some of it is friction, but this temperature forecast is your diabatic heating. That's the radiation and latent heat release, etc., etc. And for a model of even 50 kilometers, if you take that horizontal grid, you can't, model 1 kilometer of cloud and radiation that plays at molecular level, aerosol level. You can't parameterize them. So how do you do that? How did scientists do that? So this is what we call as a model, actually. So how do you solve this, in an iterative process? And all of us know that extreme events are increasing, and a lot of it has been attributed to this greenhouse gas, increase in greenhouse gas. Of course, land use, land cover, some of the work here done by Boyaz and others also show. We show that both type of effects are there, essentially man-made impacts, but here we are talking of greenhouse gas effects by contributions by Manabe and all. And as a background information, the temperature in the atmosphere decreases and at a rate what is called as lapse rate. And then this is tropopause in mid latitudes about 10 kilometers. For us, it's about 14 to 18 kilometers. Then a small, pause, tropopause, and increasing temperatures in the stratopause up to 50 kilometers because of the presence of ozone. We all know this. By Manabe's work, this Manabe and Moller's work in early 1960s, by the time people knew the characteristics of individual gases, CO2, O3, water vapor, etc., etc., but nobody thought that, thought of combining them and putting them in a model. So climate was more of a geographical problem at the time. So weather prediction for Manabe's work also started as to use, generate simple modules for weather prediction to be simulated on the computers. Anyway, so this is the first time they have combined all these mathematically and came up with the radiative equilibrium. What is radiative equilibrium? Shortwave radiation from the sun is coming, infrared radiation from the earth is going. So then at some point it goes to equilibrium, as they have shown that. And what they found is, the equilibrium, this is the observed standard atmosphere that you see the temperatures. But what they have done with the simple calculations, they got this troposphere, and then there was no tropopause, but there they have got this stratosphere. Importantly, the whole place was cold in the mid-troposphere. Okay, and the temperature was relatively warm. But in the troposphere and upper troposphere and tropopause, it was very cold as compared to observation. Okay, even less than what we call as dry adiabatic lapse rate. I'm not going into the details. It's 6.5 degrees per centigrade. So quickly, the solar radiation, sun emits radiation at 6,000 Kelvin, and Earth emits at 288 Kelvin. And based on the physics laws, Planck's laws, and Stephen Boltzmann's law, etc., we know Wien's law. We know that thermal radiation, what is emitted from the Earth, is major important factor as far as the climate is concerned, is 4 to 50 micrometers. Okay, so this is where the action is. So, and the solar radiation is mainly what we get is invisible radiation around 0.4 micrometers. So then what happens is, the greenhouse gases are transparent, or whatever gases we pump are there in the atmosphere, CO2, H2O, O3. They're transparent for the to the solar radiation that is coming at visible radiation. However, the outgoing radiation is trapped by CO2 around 15 micrometers, and water vapor has 2 bands, absorption band 6.3 and 15. There is an overlapping, etc., etc. So Manabe, by some information was there, and they combined them. They came up with the mathematics of that. At any level, the rate of change of temperature associated with this one, they have taken it as upwelling and downwelling, long wave radiation, short wave radiation. They have combined them and used the contributions from each gas. And the mathematics was formed in this way. They have this frequency, different frequencies, and combining them, total radiation, diffuse radiation from the surrounding areas, etc. They proposed this concept. Okay, it was now many physics people, our MSc meteorology people study this atmospheric physics, but it was the first one at that time. They started that. Anyway, then Manabe and Strickler introduced, it was cold in the troposphere. They learned that, they realized that it's the missing water vapor. So Manabe has proposed the concept of the latent heat release, and they have come up with this one, a simple scheme to adjust the, incorporate the water cycle. So, more of the, latent heat release and all. So by doing that, they could correct the troposphere height to 13 degrees. And another important thing that they have done is this importance of ozone. If you see this picture, the middle picture, if without ozone, the profiles were continuously cooling around 100 millibars is your trope, there is continuous cooling. But with this one, from incorporation of ozone, they could simulate this stratospheric temperature profile in the vertical. Not only that, they have also made simple experiments by simple computations by adding water vapor to represent clouds at different heights. And they have come up with this one, the proposal that, high clouds trap the heat and warm the atmosphere, lower clouds heat that, cool the atmosphere through simple reflection. So this is a path-breaking result, this too. And not only that, in 1965, Manabe at GFDL and his boss Magaronsky, they came up with the first of complete atmospheric general circulation model, as against a simple regional model like Phillips and all. So in this model, they have found out the global circulation, overturning circulations, and the transfer from the tropics to mid latitudes to the eddies, what you call as the synoptic disturbances and all these things. And you can see the left hand, the bottom one is the observed and top one is the computed climatology. Very reasonably, they got the mean climatology distributions. And the convective adjustment scheme, what I have been talking about, is a path-breaking thing and many of the models even now follow simple physics like this. I mean, similar philosophy. So what we do is, basically whenever air parcel is going up, it cools. At some point, if the lapse rate changes, goes to a critical lapse rate where it's getting to be warmer, then you release the heat to the atmosphere. And as you go up, what happens is, the water vapor, as temperature goes up, the water vapor condenses, then release the water vapor into precipitation. So he came up with this concept, a simple, now we think it's very simple, but they came up with the concept and they put this one. By doing this, what happens is whenever there's the water vapor release, it sort of warms up, latent heat will warm up, okay? The middle atmosphere. Previously, as we know, the middle atmosphere was cooler in the first few studies, but here they have adjusted that. They showed that middle atmosphere is warmer. And this cycle of importance of water vapor latent heat, they could do that because in 1967, they also fixed that. They realized that relative humidity doesn't change. Relative humidity is a ratio of vapor pressure by saturation vapor pressure at that particular temperature. As the parcel is going up, your temperatures are decreasing, and at some point it becomes saturated and water vapor has to come down. And so putting all this one, this was a game-changing paper, Manabe and Weatherall, because apart from the basic physics from the point of moisture recycling and warming of middle latitudes, middle atmosphere, they doubled the CO2 content in the atmosphere in this paper. And they found that their experiment showed that by doubling, they have got the temperature rise of 2.36 degrees. This is what you nowadays, in many models also, you see in some of the scenarios of increasing CO2, including our own Earth system model, Indian model, it has 2.36 degrees centigrade. Incidentally, I was playing a role in the first version of the model was developed by my group. I mean, I led the group, and then further developments have taken place. 2 more modules, aerosol, etc., has been added, and we have done a lot of simulations at IIT Pune. People have done that, and they have first time India has contributed, and it's one of the good models from that's the contribution of IITM. Anyway, coming to Manabe and Weatherall's work here, you can see that the CO2, 150 parts per million to 300 parts per million. These are the profiles. And in the bottom, you may not see the values, but this is where you see that 2.5, 3 degrees centigrade and all. So this is what now we do this with complex global models. This model was a simple vertical 1-point model that they have done. Okay. Then in 75, Manabe and Weatherall have doubled the CO2. And this is a total 3-dimensional atmospheric swamp model. So they have used complete physics of the model. And this is a continuity equation, thermodynamic equation, radiative equilibrium, and all, and swamp ocean model. So ocean was not, ocean currents were not there, but other physics was all there. What we still now use this to see the effects of ENSO and everything. This is what we do. And in this work also, they have demonstrated conclusively that doubling of CO2 increases the temperature. Not only that, but the hydrologic cycle of the atmosphere becomes faster. So whatever I have told about, the increase in the temperature, as the temperature goes up, the moisture, condenses, what we know as Clausius-Clapeyron effect was very much there. In their model simulations, globally average temperatures, you can see that in the surface, all this yellow is increasing, and the blue tells you that it's cooling. And this is what is observed in climate change. And this is the first study of that type. And then many people now do hundreds of simulations, and many groups do that. And because of this, the policy, IPCC, UNFCCC, etc., etc. And now the recent report tells you that definitely now we have, in the last 1 century or so, we have gone up by, last 70 years, where temperatures have gone up by 1 degrees and 1.1. And this is mainly because of greenhouse, increased anthropogenic interventions. And there is some cooling because of our aerosol radiation also. Again, these all thanks to Manabe's contributions. Okay. Manabe has not only talked about this climate change, the fundamental physics at a simple level. His concept was keep it simple. So because at that time computational powers were very less, so that's how he has developed. And even now we do the radiation parameterization and cumulus convection, etc. Even still, they are basically the philosophy, same philosophy is done. And he has contributed a lot to the other sciences. For example, he's the first guy, Manabe, Han and Manabe, they have demonstrated the importance of Himalayas. The presence of Himalayas, if you remove them, what happens? How the monsoon weakens. What is the role of the Tibetan high? And all these things were done by them in Manabe, Han and Manabe in 1975. This is the paper that has demonstrated by removing the Himalayan topography, monsoon will weaken. Another big contribution is the thermohaline circulation. In the ocean, you have this ocean conveyor belt or meridional overturning circulation. You can see red color and blue color. Blue color is the bottom, and red color is the top of the ocean in the upper layers. Anyway, so this whole conveyor belt takes about a few thousands of years to, in time scales of thousands of years. And this plays a major role in warming up the middle latitude continents, etc. And Manabe has explored, made some experiments in 1991 and 1999. What happens if you increase the CO2? What is its effect on the thermohaline circulation? Ocean circulation is not only thermal, but haline, based on salinity. It changes the density. So a combination of this, how is it affecting? So increasing CO2 through various arguments, they say that ocean freshens and then it affects the slows down your thermohaline circulation. Okay. These experiments, Manabe's hosing experiments, are very famous, and their results demonstrate the slowness of that using model simulations by changing the CO2 content variations. And and what else? Importantly, they also say that the CO2 doubling, if it is slowed down, the rate of change at which your CO2 increases, if you slow it down, what happens is the net effect at the end of the day will be more than what if you dump the CO2 at one go. So these are very complex studies, but very successfully, very conclusively, they have shown these sort of results. And Manabe has also worked on one of the biggest climate drivers, what is called as El Nino. We don't have the time. It's once in 2 to 3 years, Eastern Pacific, which is normally cold, it becomes warm once in a while in 2 to 3 years. This is called El Nino, and whole convection and everything changes here. And India, Australia, and many countries suffered droughts. And La Nina, the exacerbated condition, gives you the opposite effect. And we introduced a different type of El Nino because of global warming, what we believe is global warming. So anyway, regardless of all this, what Manabe's argument was, in one of his 1998, there were a couple of papers. In one paper, he demonstrated that the late 90s, when people said that El Nino frequency is changing, slowing down, and it may be because of global warming, he said it's not necessarily so. It can be natural variability. He went back to paleo data and paleo models. He always cautions, if you don't attribute everything to climate change, but you have to look at the past. That's the analog for the future. Anyway, that's one thing. But later on, again in this paper, they said that yes, it can be also, part of it can be because of global warming also. And importantly, Manabe's decadal variability in the tropical Pacific. He said that tropical drivers can also play a big role in the decadal variability. It's a very, very important work, which personally motivated me. And fortunately, he was there in Frontier where I was doing my Jamstack. He came to Japan also. He headed one department for a few years, went back and came back. And at that time, I had the chance to work with him, and I demonstrated the decadal variability of the Indian Ocean dipole. This is another type of coupled phenomena. You can see Manabe's. So this was I was guided by him. So Manabe's contribution is not only doing science, and he encouraged many young people like us and do cutting edge science. And like Krishnan in IITM and Professor Keshavamurthy, a lot of contributions to the Indian climate work also came from Professor Manabe, either directly or indirectly. And another work from University of Hyderabad we did is, this is also similar to Manabe's Manabe's decadal variability type of work. And all these models, what are models? We talk of any prediction. This is again from us. But all these models, global models that we're talking of, decadal prediction, that is short-term climate change. They all are basically, we all owe it to Manabe's work. And he has been contributing a lot, and he continues to contribute. Even now, he goes and quantifies, climate change. Recently, there was a paper about how much the atmosphere has warmed. There's a lot of uncertainty. So he continues that, and he proposes many, many new ideas, and only young people have to take it further. And from his Princeton page, if you see that, I'm not going through, he talks of, long-term research on global warming and model development. And he also tells about, importance of understanding past climate. And he was born in 1931 in Ehime Prefecture, PhD from University of Tokyo. Then he moved to US after postdoc and, for a job and a lot of prizes. And quickly, I have only 1 or 2 minutes, I guess. So I'll quickly talk of Hasselmann's work. Hasselmann came up with the concept of stochastic climate models. Weather is a stochastic phenomena. It's sort of in a Brownian motion. There's very, very small particles that go very randomly, whereas climate is more of a random one. So in 2 papers, Hasselmann first proposed a stochastic model and an application to this to the sea surface temperature prediction. He said that, even though the weather, winds drive the sea surface temperature, so though the weather is stochastic, a short-term scale atmosphere, you can predict slow evolving sea surface temperature. Because of that, this is a fundamental big discovery based on which people started predicting El Nino and everything later. This is a philosophical work, of course, but using real models, people have started predicting. Even of course, Hasselmann has done through some modeling studies himself. And his biggest contribution is detection and retribution. So by, whatever we say, by force so much of gases and all this type of methodology can be attributed to Hasselmann. Okay, I think I met my time, I guess. But I can't, there is a very wonderful video of Manabe if you go to your Nobel Prize webpage, you can see Manabe's interview from the webpage. He talks of how the weather had changed, climate has changed from weather prediction has changed from art to science. And he tells sort of an advice that he gives is, without understanding however complex, the complexities of science, however complex we may do, what are powerful computers we use, without understanding them, if you give a prediction, it's as good as a weather, astrologer's prediction, sort of. And this is his recent photo post Nobel Prize in a party that they have taken. And I think I'll stop it here. Unfortunately, I'll not be able to cover Maurice's work. It's beyond my domain. I hope I have done some justice to Manabe. If there is something, an issue, I'm really, my apologies in advance.
Yes.
Thank you.
Thank you very much. Now the forum is open for questions or comments. Dr. Mahadev and Professor B.J. Rao. Dr. Mahadev?
Professor Ashok, that was a very nice, wonderful talk. In fact, I'm not seeing you these days. Otherwise, I would have asked the same questions when we meet at our Venkatrao tea shop, because you moved to a new building. Yes. Anyway, this is a layman language, nothing to do with your noble talk, since you are from the atmosphere background, Austrian background. So that's why curious to know rather.
Sure.
Recently, we have been seeing the floods is happening especially in the Andhra region and Rayalaseema and Kadapa and Tirupati. So why do you think this is happening, especially in the month of November, December? So what do you think? What's happening?
Good question. The answer is also it's slightly long-winded. I hope you don't mind. First question is this November. The monsoon, one is, though the monsoon, we say June, July, August, September, there is always, we also see that in conjunction with your northeast monsoon. Immediately after the southwest monsoon, northeast monsoon sets up, it affects places like Rayalaseema and all. Okay, so there is always rain there. So second is, now what we see is, the cyclones and all. And for example, whatever we are facing today in Vizag, November is the time for cyclone season, tropical cyclone peak. And third is, extreme rainfall events that are happening in cities. In Chennai, that has happened, and in Hyderabad. In fact, one of our PhD students, Dr. Boyas, has recently got his thesis last year. His thesis is about extreme events, urban events in in extreme events in South India. We believe that there are 2 or 3 factors. Apart from one is the seasonal cycle. I think that answers why rainfall in Rayalaseema and all. But during the monsoon season also, we're getting extreme rainfall events. One we believe is because of the increase in extremity is beyond doubt. It's there. And our studies and several other studies show one is your global warming, whatever due to greenhouse gases. Second is, as Boyaz's experiments, model experiments show, land use, urban land use, land cover is changing. In urbanization has increased in Telangana and Tamil Nadu, and to some extent in Kerala, in all these 3 states. That has contributed. When you change the land use, land cover, the what you call as the boundary layer heights change, and moisture is available more than.
All right. Next question. Next question.
Yeah. Thank you.
Yeah.
Next question, Professor Vijay Rao, our Vice Chancellor.
Ashok, are you able to hear me?
Yes, sir.
Wonderful presentation. I have a simple question. Manabe's model looks extremely deterministic, which is very, very counterintuitive for atmospheric predictions.
Right.
So is it because he is looking at that kind of time scales that it has to be deterministic, or is there anything very particular about his physics?
Yeah, very good, sir. Very good question. By the time Manabe started, Manabe's idea is a physicist at heart.
Yeah.
And, he was doing development and preparing this. At that time, the concept of this, stochasticity came with Hasselmann, as you have seen that. About 10 years after Manabe's major works. So, but Manabe also realized that when he was in our climate change, frontier, by the time this, what we call as ensemble prediction has taken off. And in late 90s, people have started proposing ensemble prediction to get this probabilistic prediction. People understood by that time. But this is the fundamental development, because by the time Manabe started, there was no issue. And in mid-latitudes, the dynamics are so simple, though the quantity is so different. Up to 3-4 days, 5 days, the predictions were quite reasonable. They were predicting verticity and all. But Manabe has brought in the total real physics to predict your rainfall, predict your temperature. So that way, that is his contribution. He was not talking about predictability, but that came up in late 90s. I'm sure he must be having 1 or 2 papers on this also, but his interest was more of physics than the skills and all. Broad scale changes on climate change or weather scales. Yeah.
Very nice. Thank you. Very nice answer.
Thank you. Thanks for the nice question. Thank you.
All right. If there are more questions, I think the audience can ask the speaker the question through email or some other mode. Okay. Now we have to go to the next topic. Thank you very much.
Thank you. Thank you, Professor.
Thank you, Ashok.
The next speaker is from chemistry, Dr. D.B. Ramachari. He is from the School of Chemistry. That is our school. Okay. University of Hyderabad. He did his MSc from University of Hyderabad, PhD from IISc Bangalore, and he has a postdoctoral student at Institute of Technology. He is a fellow of the Indian Academy of Sciences as well as the NASI, National Academy of Sciences. Okay, and he has guided 70 students, and then many of them have got the prestigious award. He had more than 7 He is also an editorial advisory board member of 3 journals: Organic and Biomolecular Chemistry, Journal of Organic, European Journal of Organic, and new addition to another journals. He works on asymmetric organocatalysis. He was one of the persons who was working with Professor Barbas Jr., who was also a major contributor for the organocatalysis. Now, I will leave the podium to Dr. Ramachari. Ramachari, Professor Ramachari.
Yes, sir. Yes, sir. Thank you, sir, for your kind introduction. Able to see my slides, sir?
Yes, we can see.
Thank you. Yeah. First, my sincere thanks to Dr. Bindu Madhav Reddy, Dr. Anil Kumar for organizing this nice event, and also my sincere thanks to VC Garu, Vijay Rao for initiating this symposium. I'm very happy and to see the Nobel Committee chosen this year Nobel Prize to the chemistry is asymmetric organocatalysis. Is asymmetric I hope we all know that in organic chemistry, to perform the reaction, we need the catalyst to induce the rate, also same time to induce the rate of the reaction and also selectivity, especially chemo, regio, diastereo, enantiomer selectivity. When you say enantiomer selectivity, it's really required because in the nature, we need only one enantiomer. Is enantiomer look like is a left hand, right hand, both are mirror images, but not superimposable. So in biological aspects, and only one enantiomer only will be act as a drug for the human body, as another one will be inactive or sometimes can act as a poison. So keeping that in the mind, in the pharmaceutical, in the medicines, we have to really worry about to make the single enantiomer. For that, previously we used to use hello. So we used to use the enzymes as a catalyst. I think it's recent. Sound is okay, sir?
Is there a noise somewhere?
Yeah, exactly.
Pradeep, he has to mute his mic.
Ram, you continue.
Yeah, thank you. So, keeping that in the mind, enzymes will use as a catalyst, but the enzymes have some kind of limitations, cannot work for all the substrates. After understanding the enzymes, we went on to mimic the organometal complexes.
There is an echo, Ramachari.
Kumar Sahu, can you mute please?
Yeah, there are two microphones in his room, I think. A speaker and microphone. Two my speakers.
Is that okay now?
Yeah, one of them you may have to switch off.
Ram, now perfect. Continue.
Okay, thank you. So, the organometal complexes is a really developed very nice manner as a metal and ligand, chiral ligands, and they developed a very good catalyst to perform the asymmetric reactions. For that, in 2001, gave Nobel Prize to Professor K.B. Sharpless and Nayori and Novels for the asymmetric oxidation, asymmetric reductions. But here is a problem is metal contamination. We have to be very careful not to have any metal leaching from the catalyst to the final drug molecule. Because if any small amount of metal is there, that can act as a poison or negative for the drug synthesis. Keeping that in the mind, the organic chemist started developing is a metal-free organic molecule as a catalyst for the development and understanding about the catalysis, asymmetric catalysis. In that point of view, that is the way it came as a small organic molecule, the minimal functional organic molecule like amines or amino acid acted as a catalyst to develop these asymmetric reactions. For that work is that Professor Benjamin List and Professor Macmillan both shared this year Nobel Prize for the development of this asymmetric organocatalysis. So here I would like to explain how this area developed. For the student point of view, they can watch how this area developed from the starting to today what we are seeing. So I divided three portions. This is the era of the organocatalysis, like a pre-organocatalysis period, or organocatalysis period, a post-organocatalysis period. In the pre-organocatalysis, I can make 1954 to 1998. In 1950s, is Professor Gilbert Stoke, he able to develop for the first understanding about the generation and understanding reactivity of the enamines. He has taken the cyclic amine like a pyrrolidine and acyclic ketone like a carbonyl in presence of Bronsted acid. He able to make the enamines like that, and he went on to investigate the alkylation reaction or acylation reaction, Michael reaction. He made the intermediate, and that intermediate he went on to hydrolysis and able to get the products. See that he is the first scientist to understand the free-form enamines and iminium species, how they are generating, how we can able to formation and cleavage, able first person understand. So here he made the kind of artificial amino acid combination like a pyrrolidine acetate. This pyrrolidine acetate acted as an artificial amino acid to create this kind of enamines to understand. I'm very happy that our university of the first PhD scholar, Professor Sri Krishna, who did PhD with Professor Govardhan Mehta, he worked with Gilbert Stork as a postdoc studies in his laboratory. I'm happy to mention that. So after that, after this early 50s studies, even in the 60s, Professor Woodward and many other chemists used this artificial amino acid combination to do Robinson reaction, Michael reaction, aldol reaction, aldol condensation like that. So in 1970s, is the Ajax from the German company, they started working on to synthesize the AB, CD ring of the steroid molecules. So they are very fashionable to make the chiral AB ring, CD ring of this intermediate for the steroid synthesis. So they able to see the importance of the Stork enamine chemistry. In the Stork enamine chemistry, they're using the artificial one. And instead of using artificial one, Ajax planned that why not using the natural amino acid? This is the natural amino acid will be the deuterium form, NH2 plus and CO2 minus. They've taken the chiral catalyst. This is the chiral one, 3 mole percent DMF. They've taken this kind of tricyclic triketone. This tricyclic triketone taken and the system has plane of symmetry. And this catalyst is able to do intramolecular aldol reaction by dissymmetrization to produce this molecule in very good yield. And after hydrolysis, they got the CD ring of the steroid and it able to give 95% yield. I'm happy to say to you that this is the first catalytic asymmetric reaction. But Ajax is not able to tell clearly. He proposed some models, but these models are not that much given convincing the way to explain the how he is achieving this much facial control and this much enantiomer captures by his models. Even 1980s, Agami from the French scientist, he also explained, but he told that 2 proline catalysts are involving, one proline to make the enamine, another proline to bring the carbonate, and this amine close proximity to control the facial selectivity. From our India, University of Madras, Professor Swaminathan, he also explained to understand this reaction mechanism in 1990 by telling the surface. He told that he is not even involving in the enamine formation. He able to tell the only crystal surface is involving to bring the acetyl carbonyl and this carbonyl to go in the asymmetric induction reaction. This model also did not give that much support to understand and explain this enantiomer selectivity. Later, in the same 1970s, Wieland-Mischer ketone. This ketone also, this is the AB ring of the steroid. This is also very important starting material for the organic chemist, where using this AB ring of the steroid, you prepared so many natural products, more than 70 natural products prepared in terpenite chemistry. And this also used a proline catalyst to go asymmetric dissymmetration of this triketone to make this chiral compound in very good yield. So, this is the 1970s first asymmetric reaction using as a without metal a simple amino acid as a catalyst, but because did not understand the reaction mechanism and that 30 years people used only this reaction to make this material AB ring, CD ring of the steroid and went on to use in natural product synthesis and did not work further. Ajax did not work further understanding about this chemistry. At the same time, in 1989, another interesting paper from the Michael Jen from the UCLA, he able to take this kind of chiral alkyl pyrrolidine, this is the chiral material, and treat with this vinyl acetate chloride, he make this amide, and this amide treated with triethyl oxonium fluoroborate, and he able to protonate this O with ethyl, and during that he made them iminium species. He is the first person to realize that these kind of iminium vinyl species are more reactive in 4-cycloids with dienzole compared to counter simple vinyl system where we acrylonitrile, acryloesters and all simple one not that much reactive. But this iminium species are highly reactive. He treated, you see, 0 degrees centigrade, 12 hours, he able to make this 4+2 adduct. And after the deprotection of this, and able to get this 4+ added with 85% E. Is a very good reaction. He is the first paper to understand the utilization of this iminium conjugated species in 4+ cycloaddition. But the drawback is he didn't work further on further development of this reaction. He is not happy for the removal of this chiral auxiliary or converting the auxiliary into the catalytic mode. This is 1989. And very interestingly, in 1998, from the Russian chemist Sherebrov from the Zelensky Institute of Organic Chemistry, he able to come out homoactivated diene species like that in 1998. He take the prenol as a diene source, and this is the dienophile source. He has taken the DPPOH, diphenyl pronilol. This you can synthesize from the proline, this catalyst, taken 10 mole%. And this one react with this carbonyl will form iminium. Then after isomerization, he got this dienes. This acted as a homoactivated diene. This acted dienophile. Went on 4+ cycloaddition after 168 hours. And after the decarboxylate to deamination, regenerated the catalase and formed the cyclohexadiene with one chiral center, almost single enantiomer. It's a really excellent paper, this. But this scientist also did not work further to develop the catalyst because this catalyst is slow. Selectivity is good, but reactivity is very slow. But he did not work further development of this reaction in this area. Well, in 1990s, in the Scripps Research Institute, Professor Carlos Barbas, he is a pioneer in the 90s in development, understanding of antibodies in asymmetric catalysis. He developed the antibody 38C2, allulose enzyme 38C2, and he went on to investigate different reactions with different carbonyl. He discovered the reaction in 1995, 96, 97 in this paper, the catalytic reaction of the catalytic allulose antibody with carbonyl generate the enamine, and this enamine react with other carbonyl, went on to Robinson allylation reaction, or aldol reaction, cross aldol reaction, aldol condensation, Michael reaction. So many reactions they discovered using this catalytic antibody. When they are working on this reaction with Robinson allylation, they explain the clearly mechanism. You see, in 38C2 antibody, lysine amine act as a nucleophile, attack on the carbonyl, will form the iminium followed by enamine, and another carbonyl will come close proximity to this enamine, goes to aldol reaction, then hydrolysis of that and will form the aldol product. So in this science paper 1995 or 97, they explained clearly mechanism with the X-ray proof that antibody 38C2 able to form the carbonyl enamine. After understanding this, in 1999, Carlos Barbas, Lerner, Benjamin List, these three people taken the common substrate like this, and they investigated the both reaction with the antibody 38C2 L-proline. Both reactions went similar manner. Antibody 38C2 with a little bit fast reaction rate compared to proline, but the almost yield and selectivity almost same, opposite enantiomer form. With this paper, they realized that they know very well about antibody 38C2 mechanism. Enamine mechanism is going on. Primary amine is involving in the carbonyl enamine formation. Same way, proline also should be able to form the enamine, and carboxylic acid should induce the electrophile. Once they understand this reaction, immediately in that month they planned. Up to now, from the Ajax Paris onwards, they did only intramolecular reaction. The substrate scope is less. Now they went on to study intermolecular version. They've taken the acetone as a nucleophile substrate, aldehydes as an electrophile, added 30 mole% of proline in DMSO. Within 4 hours, they able to get the aldol product with a decent yield and up to 96% enantiomeric excess. With correlation with their antibody catalysis, they explain the reaction mechanism clearly. Because the acetone taken excess and proline reacting with the carbonyl of the acetone form the enamine like this. Now incoming electrophile, it can come the same side of the proline or opposite side of the proline. If it comes same side of the proline, this H can go hydrogen bonding with this carbonyl because of carbonyl have high electron density because of polarization. So electron pair of the carbonyl will have hydrogen bonding with the H, and this interaction, this hydrogen bond inducing the electrophilicity at this carbon, and it is going aldol reaction very smooth manner with high enantiomer selectivity manner. This is a repassive selectivity. Then this published in 2000 from the Barbas group. And immediately they went on to go study on the hydroxyacetone with excess amount taken. Yeah, hydroxyacetone also will given high regio selectivity, high diastereo selectivity, single enantiomer from the product. And they published it in 2001 full paper also with thorough investigation of the catalyst, different amino acid, different solvent, and reaction mechanism, control experiment. They thoroughly explained the reaction of this proline catalyzed intermolecular alkyl reaction. And then immediately the Barbas is a very fashionable understanding about the different reaction using proline. He is taken the nitrostyrene, how act as a Michael acceptors. Went reaction very nicely, but that E didn't come that much. But immediately he able to plan the other catalyst instead of carboxylic acid. That carboxylic acid converted to this kind of marpolin ring on that backbone and went on to study in THF. Within 3 days, able to get the very good yield and up to 78% of the Michael adduct he able to understand. So not only aldol reaction, this chemistry can go to Michael reaction also he proved in 2001 paper. And by the time Benjamin List became an independent investigator in the Scripps Research Institute, and he also started investigating other reaction like a Michael reaction. He also didn't get much EE in this. In the same 2000, he became assistant professor in Scripps, and he able to come out the Benjamin List a 3-component manic reaction and the proline catalysis. Is a manic reaction is one of the very, very important reaction to produce the chiral amines, very important molecules in the drug discovery. And he has taken the incidin, aldehyde, aromatic aldehyde, will go ship-based formation between these two, and the proline will react with this acetone, substituted acetone, will form the enamine, and this is the transition state, he proved it. And here is the PMP will this side and RL this side will go C-facial attack to give high enantiomeric access. This is the Benjamin List published in 2000, 2002, will full paper explain thoroughly this 3-component reaction. Also, same time from the Barbas group, they have taken the different substrate based on the ethyl glyoxal imine species. These are preformed imine species they have taken and taken the aldehyde as substrate or ketones as a substrate using L-proline. They prepared the Michael adducts almost single enantiomer, single diastereomer. These species are very, very important molecules in the discovery of the artificial amino acid. This ester one, if you make hydrolysis, this if you deprotect with the copper catalyst or DPP or system, whatever MPMP, you can get the artificial amino acids or functionalized amino acids. This is a very important paper in 2002. This is back-to-back paper published in the JACS from the Carlos laboratory. So immediately Carlos, here is forming the syn product, right? Immediately design the catalyst, other catalyst to make the anti-manic product in a very good yield, and he explained how this OME is facilitating to make this enamine formation. Here is enamine is forming anti-enamine. Carboxylic group and enamine is anti, but here is this enamine and OME is a syn. Very interesting phenomena he observed to explain how to make the anti-manic adducts in very good yield, and he able to study water effect also. Small amount of water is facilitating excellently high rate reaction, high rate and high selectivity. Very nicely demonstrated the water. He also, because he works on biology and antibody catalysis, he believed that organic chemistry also, in the organic laboratory also, reaction should go in the cellular mode. Should not unnecessarily stop the intermediate reaction, isolate, do unnecessarily column chromatography and all. Why not performing in the cellular mode is to believe. That's what he makes to make the asymmetric assembly reaction. He made the proline catalyzed manic reaction, followed by addition of the diethyl aluminum nitrile in one part. He able to make the cyanation chemistry also. And by just changing the temperature, he went on to cyclization with high enantiomer selective mode. In the Benjamin List, by the time I think he moved to the Max Planck, from the Scripps to Max Planck, and from there he started investigating the importance of this organocatalysis and other reaction. He has taken the CN bond formation. You see that this is the new CN bond formation. He has taken the immunium species like that diazo species and he able the asymmetric catalytic amination chemistry. Almost same time from the Denmark, Professor Jorgensen, he also developed this amination chemistry using the aldehydes or ketones as a substrate with this diethyl isodecarboxylate as a amine source. And we are very happy to mention that in this paper, Professor Basavaya given PhD Gumaraburgan, he went on to Jargonson and he developed this chemistry, entire this chemistry. Kumaraburgan developed, who did PhD in our School of Chemistry. This work. Almost these two papers came back to back in JAX. At the same time, the Carlos understood the one-part chemistry. He has performed the emanation and aldehyde has taken aldol reaction to make highly functional molecule, where he went on to show the medicinal application of this molecule as a renin inhibitor. And the Benjamin List from Germany, he able to understand the importance of this reaction. He has taken the terminal dialdehyde like this and added the proline, went on to make the cyclic chiral alcohols in very good enantiomer selectivity. You can see here. This is the way approach. We know the enol endocyclization, but he designed the enol exocyclization, how the proline can able to control. This is the Ajax type model. H is controlling the aldehyde like this, hydrogen bonding. If it goes like that, whether this O can able to go hydrogen bonding with H or not. He designed this reaction. He demonstrated beautifully how nice 10 mole % of proline can able to do intramolecular cyclization to make high enantiomer selective cyclic alcohols. And 2003, this is one of the excellent year for the further demonstration of the organocatalysis. Benjamin List with collaboration with K. Nuck from the UCLA, he went on to do quantum mechanical prediction of the stereoselectivity of the proline catalyzed asymmetric intermolecular aldol reaction. They went on to study variety of the conformations, rotations, vibration, all they have studied. And finally they found that proline, this is the way is going to form the enamine with the carbonyl, and incoming electrophile will come like this. R group is that side, H is this side to prevent the repulsion between this group, and H is involving hydrogen bonding like that. This is the diagonal angle, bond angle, all they investigated to make this is the lowest energy pathway to make the high enantiomer selectivity. After this paper, and the same group went on to investigate the kinetic and stereochemical evidence for the involvement of only one proline to involve in this transition of proline catalyzed reaction. Previously, 1970s, scientists believed the two prolines are involving. But in these two reactions, Benjamin List proved that it is only involving only one catalyst by this retroaldolization reaction, by changing the proline E loading. They didn't observe any nonlinear effect. By changing the dilution, they didn't observe any nonlinear effect. By changing the dilution in the reaction, changing the proline EE percentage in the reaction, studying the retroaldolization, nowhere they observed any nonlinear factor in this reaction. From these two papers, Benjamin List and K.N. Huck both proved that proline is involving in enamine mechanism, is only one proline involving in the reaction. They proved it. After this, and the Benjamin List came out that he able to prove the intramolecular alkylation reaction. It's a big challenge actually. If you take the catalyst and if you do intramolecular alkylation reaction, HI will form. That HI in situ generated acid can prevent the nucleophilic nature of our organocatalyst. For that, he has taken triethylamine, one equivalent, and this is the catalyst, and he able to demonstrate excellently intramolecular alkylation reaction with high enantiomer selectivity. He proved with the different substrate to show the sustainability of this reaction in a very good manner. Sorry. He also demonstrated the other organohydrogenation reaction, asymmetric hydrogenation reaction, organohydrogenation. He involved in the tandem approach also. He proved the mechanism in two back-to-back papers in Angevante JAX. He proved how important organocatalysis for asymmetric hydrogenation reaction. The way the Nayori went on to make the hydrogenation reaction with these kind of spaces without metal catalyst, able to demonstrate how simple organic molecule is able to catalyze the asymmetric hydrogenation reaction to make similar molecules in very good EE. At the same time, in 2004, Carlos Barbas, normally in organic chemistry, we generating the quaternary carbon is a very difficult. Sometimes we have to go very reactive catalysts and different conditions to make the quaternary carbon. But Carlos, very simple, he designed the catalyst like that and able to make the co-catalyst, TFA, able to generate the quaternary carbon with high enantiomer selectivity and diastereomers selectivity, and went on to aldol reaction also with generation of the quaternary carbon. He make with the simple catalyst, he able to demonstrate that he is the first paper to demonstrate that quaternary carbon can generate through organocatalysis. And at the same time, he has taken the simple substrate like alkyl benzylene acetone with different aldehyde and cyclic system, able to demonstrate the homo-activated 4+ cycloaddition reaction to make the highly functionalized 4+ cycloaddict in single diastereomer, single enantiomer. Later, so many people worked all over the globe on this reaction, and we wrote the textbook on this reaction also. And now people are calling this as a Barbas 4+ cycloaddition reaction. And this is a story from the Benjamin List and Professor Carlos Barbas from the Scripps and MIT, Max Planck. At the same time, from the Berkeley, from the Caltech, from the Princeton, he worked on 3 places, Professor David Macmillan. In 2000, first he is from the Berkeley, he developed the reaction very nicely. He synthesized this imidazolone catalyst from the phenylalanine catalyst, and he developed this imidazolone catalyst with HCl as a co-catalyst. He has taken this alpha-beta aldehyde as a substrate and cyclobutadiene or acyclic diene as a diene source. In methanol water combination at room temperature, he able to get the 4+ cycloaddict. This is a LUMO activation. This catalyst will react with this carbonyl, will form immunion species like that. Once these immunion species are formed, and as the Michael Cheng proved that in 1989, the immunion species are more reactive compared to corresponding neutral species. This immunion species highly activated. Now the top phase is controlled, covered by this benzyl group. Now incoming diene source will come only from the bottom side. And that facial control is there more than 90%. That's why you're able to get more than 94% highly enantiomer selective cycloaddict. Only the DR ratio is not that much good, endo-exo, but enantiomer selective is very good. Immediately in coming years, he proved how important this catalyst in 1,3 dipolar cycloaddition reaction with the N-oxide species. Then immediately he demonstrated the reaction with the Friedel coupled alkylation using the pyrroles as a nucleophile source, and immunion species as a Michael acceptor. He went on to investigate the excellent product formation with high enantiomeric excess. He demonstrated with indoles as a Michael donors, and this is a Michael acceptor. Anilines as a Michael donors, and this is the Michael acceptor in different Friedel coupled alkylation reaction. By the time he synthesized the 2nd generation catalyst, this is the 3rd generation catalyst. And this we used the enones instead of aldehyde. This also chemistry went very excellent manner to achieve high enantiomer selective 4+ cycloaddict. In 2002, he able to understand the Carlos Barbas list aldol reaction, and he came out the cross aldol reaction between sterically crowded aldehyde and less crowded aldehyde. Less crowded aldehyde became a nucleophile, sterically crowded aldehyde became electrophile. This is the transition state, and it went on to make the aldol products like that very good selectivity. He has used his catalyst also, his own catalyst as a substrate instead of proline. He went on to the functionalized aldehyde, especially O-protected aldehydes, and went chemistry very nicely. He prepared the sugar intermediates, sugar molecules, and published it in JACS and Nature in 2003, 2004. He demonstrated organocatalysis as a chiral amino acid, how suitable catalyst to prepare the sugar molecule he demonstrated. I didn't cite that paper here. I think you can visit his website to see the excellent work of the sugar synthesis from the amino acid or amine catalysis. At the same time, he able to develop the new reaction where proline catalyzed asymmetric oxidation chemistry. You can see he taken the nitrosobenzene and went on to make the asymmetric oxidation, and he demonstrated the reaction mechanism. Almost same time from the Carlos group, he is assistant professor from the Professor Carlos. Same time he also developed this reaction, and almost same time published in Angevin Jack's paper the importance of this organocatalytic asymmetric oxidation followed by aldehyde reduction chemistry. It proved the reaction mechanism very well. Also, same time from Macmillan, he used this his catalyst and able to make the using this substrate as a asymmetric halogenation reaction. What I want to say is in Benjamin List, Macmillan, Carla, they able to develop as much as understanding about this enamine, catalytic enamines with different bond formation, whether it's halogen or oxidation or emination. Very beautifully chemistry went very nicely for high enantiomer selective reaction. And up to 2004, from 1999, Professor Carlos Barbas, Professor Benjamin List, Professor Macmillan, Jorgensen, able to demonstrate in different reactions, understanding about generation, catalytic generation in situ, and application in aldol, Michael, Manic, emination, Diels-Alder, oxidation chemistry, and all. So after the 2005 onwards, the many people entered in this field. I can say 1999 to 2004 is the real organocatalysis period where expanded the subject, and 2005 onwards, many people entered. But still, the Professor Carlos Barbas, he developed so many aspects, especially asymmetric assembly reaction, total synthesis application in this organocatalysis. He developed the phosphine catalysis reaction. He developed the amide donors for organocatalysis asymmetric reaction, and 3-cycloaddition, hydrogen bonding catalysis, and especially anti-selective asymmetric manic reaction. He able to develop acyclic amino acid as a catalyst. I would request to visit his website to see how many papers he demonstrated, explained to develop this area in the bigger level. At the same time, Macmillan, he also developed the organocascade catalysis after 2004 onwards, and he applied in total synthesis and reduction, oxidation, epoxidation, especially amine conjugated, especially developed the SOMO activation, singular occupied molecular orbital catalysis, and he merged the photoredox with the organocatalysis. These two areas are extraordinarily developed the organocatalysis in the further level, and he demonstrated the catalyst as a commercially available catalyst, the first generation, second generation, third generation. They all catalysts are commercially available. And please visit his website to see further how he is contributing the organocatalysis. And Benjamin List, he also developed from the Max Planck a powerful bronze acid catalysis he developed. You can see here bronze acids. This bronze acid can use very catalytic amount you can load to have different bronze acid catalysis reaction, asymmetric reaction. And he able to develop counter-anion directed catalysis. And he able to develop the asymmetric applications in total synthesis and also oxidations. I would request you to visit his website to see further development of organocatalysis. Yes, Jorgensen, he able to develop Michael emination, and especially is an homo-activated 1-amino-methylbutadiene. He developed so many reactions by using dienamine, trienamine, tetraenamines in different reaction. I'm very happy. And he able to develop the asymmetric oxidation reaction, especially epoxidation reaction, where the Sharpless used allylic alcohol, he used the cinnamaldehydes as a substrate to make the epoxides in using metal-free catalysis. Very beautiful work he developed under organocatalysis mode. And also from the Ayashi, from the Japan and Jorgensen, this is the Ayashi Jorgensen catalyst. This is the catalyst with OTMS, DPP OTMS. They developed, used this catalyst as a enamine chemistry or iminium chemistry. You can see, taken the aldehyde and nitromethane addition and a simple transformation, able to make the baclofen and pregabalin. These are the drug molecule in the market. And the Japanese company able to utilizing this organocatalysis to produce these drug molecules. You can see how simplified. There is no metal. Once you get the drug molecule, you don't need to worry about the metal contamination in this drug molecule. You don't need to worry. Even ppm level or ppb level, you cannot find any metal. That is the beauty of the asymmetric organocatalysis. And Jacobson from the Harvard, he developed the 2002 onwards a beautiful catalyst, the thiourea catalyst, to use a very low loading at low temperature to make the Stecker reaction and hydrophosphorylation reaction, manic reaction. You can see the product, how energy selectivity able to produce. And Taibatsu, he developed the catalyst of synchronoalkylide with thiourea, one of the beautiful, very, very useful catalysts in many synthetic transformation in organic reactions. Nowadays, we are using that. And the Viresh Raval from the Chicago, he developed the hetero-Diels-Alder reaction in hydrogen bonding catalysis. Excellent work he published in that aspect. And he synthesized his catalyst. We'll call it, this is a Raval catalyst. This Raval catalyst, coaromatic catalyst, is designed and synthesized. It demonstrated how important this chiral square catalyst in Michael Mannick reactions. And also Enders is one of the pioneer in organocatalysis, especially in NSC catalysis. He demonstrated how important organocatalysis in cascade chemistry. You can see these three substrate he has taken and used the DPP-OTMS catalyst, able to synthesize the drug-like molecule in just one part. This is the chemistry. This is the catalyst. Between these two substrate will go iminium chemistry. You get the substrate. You go to the iminium catalysis to go to the next further reaction, and you'll go to intramolecular reaction, and catalyst will regenerate and product will form out. This go high catalytic demonstrated, published in 2006 Nature paper. And Shugawa from the Germany, she able to design the excellent catalyst, the bifunctional catalyst, to demonstrate the both are there. Nucleophile, electrophile nature is there in the catalyst, able to go asymmetric Michael high selectivity. She also demonstrated excellently autocatalysis concepts in asymmetric organocatalysis. Blackman from the Scripps, she able to utilize the kinetic analysis of this chemistry to understand what is the primary catalytic species, a secondary catalytic species, or you can say as a parasite species. These parasite species, how controlling the organocatalytic reaction rate and selectivity, she explained thoroughly using the reaction process kinetic analysis. I request you to visit our website to see the importance of this work. This is the way it is going on from the 2005 onwards in outside the country and organocatalysis. Here I would like to share what is happening in our country in the organocatalysis area. As I told you, Professor Swaminathan from the University of Madras, in 90s he worked on proline catalyst intramolecular reaction and the same kind of Ajax-Paris type reaction. But he didn't believe that proline is able to make the enamine species to go intramolecular reaction. He believed that crystal surface is involving in catalytic asymmetric reaction. And another professor from our school, Professor Baswega, he in 80s he taken one of the patent like a tertiary amine catalyzed CC bond formation between enones and aldehydes as the mark the Bailey-Silman adduct. Bailey-Silman they published in patent in 1980s. That patent he has taken and he rediscovered the importance of that reaction, solvent, catalyst, and application. He wrote almost 3-4 reviews and a lot of papers he published. The way Ajax-Paris reaction, the way Barbas and List, how they rediscovered. And Professor Baswaya, he rediscovered the tertiary amine catalyzed aldol reaction. And all over the globe, many people use it today. This is the importance of this Bailey-Silman reaction in organic chemistry. Other scientist, Vijay Nair, and he developed the NSC catalyzed reaction, how important the N-nitrocyclic carbene. I would request you to go through his website to further understanding about his discovery. And Professor Vinod Singh from IIT Kanpur, he developed his own catalyst like this. Instead of 1H, he taken the two kind of amides catalyst like that and went on to do reactions in aldol reactions.
Professor Ramachari, how much time do you need?
Another 10 minutes.
10 minutes is difficult, Ramachari.
No, I have given 45 minutes. I asked the organizing committee to give me 45 minutes.
Yeah, that 11:45 got started.
Yeah. Another 10 minutes. Thank you. So is Vinod K. Singh, he developed this reaction very nicely in water mediated chemistry went to make the aldol product. And from the Germany, Brussels, he produced this catalyst in the large scale. This catalyst produced. Today, we all over the globe call it this is Singh catalyst. And Pradeep Kumar from the NCL Pune, he utilized the organocatalytic amination reaction in total synthesis. And Shantan Mukherjee and Shubha Chandrapan both did PhD with Benjamin List from Germany. They from Mukherjee from the Bangalore. He is working excellently thiourea catalyzed asymmetric reaction, and he also developing functionalized molecule synthesis. He is the Indresh Kumar who did postdoc with Ujiro Ayashi from Japan. He joined BITS Pilani. He able to demonstrate how important this Barbas dienamines in 4-pesocycloaddition to synthesize highly functionalized chiral molecules. And I can tell so many young chemists are working in our country, in especially asymmetric catalysis. Last year, we wrote the review on who and all working in asymmetric catalysis in the last 1 decade. especially Professor Jain Murthy, V.K. Singh, Chimini, Professor Peresami Garu from our School of Chemistry worked extraordinary. Whatever today I am telling about this all chiral organic amines and all, he synthesized so many chiral amines from the resolution tool or is from the reduction. He has the Peresami reduction condition used to make the amino alcohols and all. I discussed so much in this review. I would request some of you guys to go through this review to see who and all working in this asymmetric catalysis from this country. I joined 2005 in School of Chemistry. My passion is to develop the sustainable reactions using organocatalysis. And we developed this three-component retroalkylation reaction. We went on to utilize in so different chemistry to apply. Especially, we developed this last one important starting material in organic transformation, asymmetric transformation. And recently, you can see how important drug molecule we are able to synthesize using LASSON, using our asymmetric organocatalysis. And last month, we demonstrated how important this ppm level catalyst loading in organic synthetic transformation and also propagation. These and all, we demonstrated the large ground scale reactions in organocatalysis to simple filtration technique we are able to apply. And that is what I can say. It's not only demonstrating, others also should utilize important. I'm very happy to tell you that Pfizer used this, our reaction, to make the hepatitis C virus drug synthesis in the kg scale and also in total synthesis. And I'm very happy to share that as organic chemist expert that. And also we developed the click reaction where Sharpless developed the copper mediated chemistry, Betroji developed the strain promoted reaction. We developed the amino acid mediated click reaction where we showed the medicinal application. And you can see we went on to show the lot of application in medicinal chemistry. And present today, I'm very happy that many people are utilizing this reaction, metal-free click reactions. And also we developed the amino-inane catalysis. We developed other phenomena is a supramolecular organocatalysis. This is the, I'm happy to say this is the fourth pillar of the catalysis where you can see this chemistry. This chemistry Barbas developed. When I put the other one, functional group reaction didn't go. And we checked so many catalysts did not go. Then we able to understand what is the importance of the supramolecular assembly generation in the pre-transition state. And we able to make the combination of the two catalysts. Chemistry went very nicely. We went on to study the control experiment and we used the online HRMS analysis to understand the pre-transition state. We proposed the pre-transition state and we further working on this area. And industrial applications are plenty, organocatalysis. You can see this is a Japanese company using, especially from the Spain, advanced cell therapeutics. This is the drug molecule. This is the drug molecule ruxolitinib, able to use the DPP-OTMS catalyzed azamical reaction. Is using the kg scale. And also recently Chemical Society review and Physical Science reviews, so both are excellent reviews published from the Peng and Carlon. I would request you to go through these two reviews to see the importance of asymmetric organocatalysis, especially in medicinal chemistry and pharmaceutical ingredients. So I'm very happy to conclude here that we are very thankful to Nobel Committee 2021 for choosing the Chemistry Nobel Prize for the asymmetric organocatalysis, who pioneered in the front line, the Benjamin List and Macmillan. Here I am very unhappy. One of the other big name is missing. That is the Professor Carlos Barbas. Is the bad time is he very early he left the planet and he went on to the Sorgalo. Maybe he's watching from there. As long as organocatalysis is there on this planet, we'll remember his name as Professor Carlos Barbas, who pioneered to introduce this new area. And I'm very happy to say that still a lot of work is there. Need to expand the library of sustainable organocatalysts and need to expand the substrate scope, need to expand the more number of green sustainable reactions through organocatalysis, especially need to load the ppm or ppb level catalyst loading as similar to engine or better than that. And also need lot of industrial application from this subject. Lot of work is there. I can say I'm very thankful to my students and especially my funding agencies, SCRB. Thank you very much for your patience.
All right, Professor Ramachari, thank you very much. Before I go to the questions, okay, I just wanted to mention that Professor Ramachari worked with Professor Barbas. It's evident from a few slides. Okay, Barbas also would have gotten the Nobel Prize, it looks like. Okay,
absolutely.
So this is something that all of you should know. Now I will go to the question and answer session. Okay, we have limited time. Madhu Babu, and VCOH. Madhu Babu?
Sir, Professor Ramachari.
Please. Thank you.
As a student of organic chemistry and biochemistry in my undergrad days, I wondered there are a lot of chemical reaction that happen in normal metabolism. They have a lot of similarity to organic pathways. So I was wondering, because enzymes are foolproof they carry out foolproof stereo selective reactions. So when do you think we will have these kind of enzymes in the industry of selective organocatalysis? Because since you were mentioning about sustainability, I believe having enzymes doing this job would be more sustainable and good.
Actually, Madhubabu Garu, that's what I want to ask. The way this is the what you have enzyme lot of so many functional group, then it is created the whatever the available area, some small area, pocket area. In that pocket only the reaction has to take place. That pocket is a suitable for only suitable substrate. That's what the limitation is there for the substrate scope. So you remove the unnecessary portion, very minimal functional group is required enough to make the asymmetric induction in the reaction. That's what prove the organocatalysis. When you have a simple small amines able to enough, then why need to go back again enzymes?
Okay.
Dr. Ramachari,
sir.
Are there some good examples of small molecule organocatalysis in living cells?
Excellent, very nice question, sir. That's what we need to, especially metabolites. The small metabolites, whether is able to involve in organic transformation synthesis in the cell, that we need to find out, sir.
Because sometimes metabolite levels go very high up.
Yes, sir. Yes, sir.
Yes, in sub-millimolar level. So there it is very likely that they may be catalyzing some interesting biochemical reactions.
Yes, sir. Absolutely. That is one of the beautiful questions, sir. We need to work out, sir.
Yeah. And one more small question.
Please.
Can we theoretically predict why some organocatalysts are very slow?
Yes, sir. Yes, sir. Because these molecules have a self-oxidation, because any air oxidation, anything if a small contamination, any impurity is able to decrease the catalytic power of the functional group, sir. That's why it won't go react. Because very sensitive, sir.
Okay. Thank you. Thank you.
Thank you.
All right. I think now we have to go for the next session. So to conclude this session, let us thank all the speakers of this session. Okay. They were very good presentations. All of you, thank you very much.
Thank you, all the speakers. I'll join Professor K.C. Kumaraswamy also to thank all the speakers in the first session. And we would like to thank Professor K.C. Kumaraswamy as well for being a chair and then conducting the session. So now I would, I'll give it to Anil to invite the second session.
We also thank all the organizers for the wonderful job.
Thanks, Madhu. We have still half left. Sir, welcome back for the second session. First of all, it's we thank to first session chair, Dr. Kumaraswamy. Thank you very much, sir, and I will also thank the speakers. This second session is still very interesting, and I hope everybody finds very curious to hear the talks by the experts. The chair, Professor Vinod Paurala, we request you, sir, to be chair, and we thanks. Dr. Vinod Paurala is a senior professor in the communication department at the University of Hyderabad. He's also hold a UNESCO chair on community media. Dr. Paurala joined the University of Hyderabad in 1995. Before his academics were, he got a dual degree in sociology and communication, and also PhD from University of Pittsburgh. Then he served as a faculty at Pittsburgh and Virginia Tech University and IIT Bombay before he moved to the University of Hyderabad. At the University of Hyderabad, he served as a HOD and Dean of Sarojini Naidu School of Arts and Communication, and is served also Director of International Affairs. As a founder president of the Community Radio Forum of India, Dr. Paurala played a significant role in drafting the national policy permitting the community radio as a space and radio dial in the country. We are glad and thank him for serving as a chair, and I request to start the session this afternoon.
Thank you, sir.
Thank you. Thanks to the organizers for inviting me to take up this responsibility. So after the first session where we had all the science prizes, in this session we are going to look at this year's Nobel Prizes in economics, literature, and peace. These, by virtue of the kind of prizes they are, tend to get a lot of public debates, and ordinary citizens usually get into discussions about these prizes because some of the work for which these prizes are given seem to directly interest some of the people, people at large. Let me start straight away with the first lecture, the first set of presentations on the Nobel Prize in Economics. Just to give you an interesting fact, the Nobel Prize in Economics is actually officially called the Sveriges Riksbank Prize in Economic Sciences, named after the Swedish central bank. So it wasn't originally part of Nobel's endowment, fairly recently established in 1968. But from time to time, because of the kind of people who win the prizes, more recently, people like Professor Amartya Sen, more recently Abhijit Banerjee and Esther Duflo, it created some degree of interest and excitement in India. This year, the Nobel Prize has been given to David Card, Joshua Angrist, and Gildo M. Benz. And the Nobel Prize committee said half the prize goes to Professor Card and the other half is divided equally between the other two winners. As a senior economist, David Card gets the major credit for this work. Interestingly, University of California, Berkeley, where Professor Card works, announced that they would give him free parking for life. And I thought that's a small incentive for his tremendous achievement. So we'll start by talking about Professor Card's contribution. Dr. Vijay, I think, is going to talk about Professor Card's contribution, followed by... Yeah, is that right, Vijay?
Yes, sir. Yes, sir. Yeah.
And Professor Naresh Sharma on the other two winners. So briefly, Dr. Vijay is currently assistant professor at the School of Economics. He specializes in labor economics. Before joining the university, he was earlier at Institute of Public Enterprises and also NALSAR, the National Law School in Hyderabad. His PhD in Development Studies was from the Institute of Social Studies at The Hague in the Netherlands. And he is also the recipient of the Young Labor Economist Award for the year 2008 from the Indian Society of Labor Economics. So well published, both in terms of his journal articles and books. So let me welcome Dr. Vijay, and let me also introduce Professor Sharma, and then they can both conduct the proceedings. Professor Naresh Kumar Sharma is one of the senior economists we have at the School of Economics at University of Hyderabad. He was formerly Dean of the School of Economics, and his PhD was from Indian Statistical Institute. His areas of work and his main contributions have been in the areas of economic theory, Gandhian economic thought, Indian economic thought, development economics, money and finance, and so on. Fairly well known in the field. I'm happy to have Professor Sharma join this. So Dr. Vijay and Professor Sharma, between you, as the organizers have pointed out, you have about 25 minutes, and then we can take some questions. So I hope you can divide the time accordingly. Over to Dr. Vijay.
Yeah. A very good afternoon. Is my PowerPoint visible, sir?
Yes, it's visible.
Okay. So, respected Vice Chancellor, the Chair, colleagues and friends, students, We are here, we're quite proud to be on behalf of the School of Economics to present about the works of this year's Nobel laureates. I will be presenting the work of Professor David Card. And as the citation goes, the Spurious Riksbank Prize in Economic Sciences in memory of Alfred Nobel 2021 was divided one half awarded to David Card for his empirical contributions to labor economics. And interestingly, Joshua Angrist was the student, actually, of a PhD student of Professor David Card at Princeton. And he, together with Guido Imbens, has shared this year's Nobel Prize. Professor N.K. Sharma will be reviewing the other two scholars. Now, to understand the contributions made by Professor Card, he has a very large body of contribution to labor economics, extending to a variety of dimensions, including wages, immigration, social protection, trade unions, technology and skills, as well as methodology. However, for an understanding, given the paucity of time, we will just stick to his analysis about consequences of changes in factors like immigration and minimum wages. Now, the academic analysis, media debates, and policy perspectives on immigration and the minimum wage policy have been contentious. They've been conflicting and controversial, and this is because of the fact that the underlying this whole disagreement and the polarized views are serious methodological limitations. Now, if you look at the national public radio-based news, or for that matter, the New York Times, we see that senior policy advisors have been thinking or contemplating on the basis of proposals to dramatically curtail legal immigration in the US. And this has been backed by contributions of academicians like George Boras at the Harvard University. Similarly, with reference to the minimum wage policy, if you look at the specialized journals like the Wall Street Journal, here is David Newmark, a professor at Michigan State University, who in 2015 has analyzed Mr. Obama's policy of wanting to enhance the minimum wages and then linking it up with loss of jobs. And again, in 2021, he writes about Mr. Trump's policy initiative to block rise in minimum wages, but at the same time talks about a letter that has been circulated by Nobel laureates, which basically argues that there is no relationship between minimum wage increases and job losses. Now, if you look at the theoretical models underlying this whole controversy, we see, for instance, on the left-hand side from the figure, that assuming that at the demand AFL and assuming that there is a perfectly inelastic supply of only the natives in the short run, you would have a wage rate of W0. And now, if you assume that there is an increase or an addition, an inflow of immigrants, then the supply curve moves to LS hash. And that leads to a reduction in the wages for all the workers, including the natives, from W0 to W1, which basically means that the natives lose their share in the national income of W0, B, W1, D. And BDC is the area that then represents the additional producer surplus that accrues to the capitalists as a consequence. Now, this theory, originally based on formulations by Professor Hicks, who was himself a Nobel laureate, George Boras at Harvard University has basically, simulated empirical data, and on the basis of which he basically says that, with the exception, of the instances where the immigrant workers and the native workers have skill differences, or where the immigrants, basically complement the native skills, or where the immigrants bring in additional capital stock, or in the instances where the social protection of the immigrants is covered by the native country or their own country of origin. With those exceptions, a general inference can be made that immigration, or especially of unskilled labor, leads to adverse effects on the wages of the natives. And based on the empirical simulation, he has suggested that immigration-induced change in relative supply of high school dropouts reduces their relative wage by 4.8 percentage points, or about 44% of the total decline in the relative wage of high school dropouts between 1980 and 1995. Similarly, if you look at the theory on the enhancement of minimum wages, we begin here with an equilibrium wages of PE, at which you will have an equilibrium, market equilibrium employment of EE. And if we assume then that there is an enhancement through a legislation of the minimum wages from PE to PR, then at that wage rate you will have a demand which is ER for employment, whereas at that wage rate you would have a supply of EU, and the gap basically represents the unemployed. And here we have George Stigler, who in his 1946 paper has basically argued, and he himself is a Nobel laureate again, the higher the minimum wage, the greater will be the number of covered workers who are discharged. And on the basis of the empirical works, it has been concluded that for every 10% increase in the the coverage adjusted minimum wage, there is a likelihood of there being anywhere between 1 to 3% reduction in teenage employment. Now, what is the problem, and why is there a controversy? Is because if you look at the conventional theory, it is based on highly restrictive assumptions: a linear homogeneous production function, constant returns to scale, competitive prices, homogeneous labor units, perfect substitutability, and so on. Now, all these assumptions in turn have lead to particular kind of evaluations regarding the elasticity of the relative wages, and eventually lead to particular kind of estimates of the impact of immigration. And quite clearly, those assumptions are problematic when compared to the real world situation. And similarly, on the other hand, those people who have disagreed with Professor Boras have come up with empirical evidence on the basis of cross-city comparisons. However, each of these cities is a different labor market, and those dynamics that operate in each of these labor markets imply that you cannot really be talking about the impact of one variable, and therefore the inferences have weak credibility. It is in here that Professor David Card's natural experiments is a methodological input that really addresses these kinds of methodological issues. What is a natural experiment? Now, unlike natural sciences, carrying out experiments in social sciences is very difficult. While social experiments on the basis of randomized trials have been conducted by behavioral economists, not all problems allow such experiments, and further, there are serious ethical concerns. And to answer big questions like, for instance, what is the effect of immigration on wages of natives, or what is the impact of minimum wages on employment, how can we really set an experiment? And here, a natural experiment involves taking an actual event which has occurred in a society, say, for instance, an actual immigration or actual increase in minimum wages as a context, as if it were a setting for an experiment, and by comparing the society affected by the event or the treatment group with an unaffected or control group, observations are made towards understanding causal relationships. Now, in addition to the natural experiment component, Professor Card also has before and after, in temporality terms, effects comparison, comparisons in his work that lay bare causal effects other than the one being investigated. So here is the first instance of the application of the natural experiments to the study of the problem of impact of immigration on native wages. During 1980, there was the Mariel boat lift, which brought about 125,000 Cuban immigrants into Miami, US. And whereas the national average, normally in the US the increase in the workforce because of immigration is around 0.3%, in Miami, it increased in 1 year by 8%. Miami, therefore, was an ideal setting for a natural experiment to test the hypothesis that entry of immigrants leads to reduction in the wages of natives. And David Card, by taking Miami as a case and comparing it with other unaffected cities for the period before and after the immigration, studied the impact of this immigration on the wages of the natives. Now, in course of the study, the entire workforce is stratified into natives, earlier immigrants, recent immigrants, and Card observes the impact of immigration on the wages by further specification of the workforce characteristics of skill, age, ethnicity, and nationality across different occupational distributions. Now, what were the major findings? And here we observe that in comparison to the pre-Mariel boat lift trend line of wages, the inflow of immigrants shows no significant impact on the wages of natives or earlier immigrants. In fact, it shows an increase. Now, what causes the average wages of unskilled workers as a group to dip is the rise in the relative population of new unskilled immigrants who as a group are receiving average wages which are much below the average wages of the other two groups. And it is this proportional share of the population which in fact causes this kind of a reduction in terms of inferring from a assumption of homogeneity. Or because in places like Miami, the relative population of the natives becomes so thin that the sample is also extremely small. And the other application of the natural experiment to the minimum wages is by way of taking the example or the case of the legislature in New Jersey increasing the state minimum wages to 5.05 per hour and comparing it with the adjacent Eastern Pennsylvanian labor market, and wherein taking this as the setting for the natural experiment, Professor Card, together with Alan Kruger, carried out surveys in several fast food stores, and he did this for the minimum wage legislation and the post minimum wage legislation, and then regardless of whether we compare the stores in New Jersey that were affected by 5.05 minimum wage in stores to Eastern Pennsylvania or to stores in New Jersey that were initially paying only $5 per hour, quite interestingly, we find that increase in minimum wage increased employment. The scale of the stores in New Jersey actually expanded in the size. Therefore, the empirical results reject the predictions of a conventional model, and alternatively, it can be seen through the empirical evidence that firms can be, seen to be price takers, or that they are essentially competitive, or there is competition in the product market while having some degree of market power in the labor market. Now, finally, to then talk about why is there this kind of anxiety about immigration leading to decline in the wages of the natives, Card explains that it is more on account of compositional effects, that because of the increase in the relative population of the immigrants, there is a worry amongst the natives, and that worry might be explained because of social cultural reasons, that their culture is being lost, or that it could be Islamophobia, it could be racism, it could be anything but lacks an economic reasoning in the way that the anxiety is flaring up. The degree of the market power of the firms is being put to a very interesting application to the study of gender and race-based discriminatory low-wage payments. This has been done by new Keynesians, especially by post-Keynesians, in what is called as the social structures of accumulation. So thank you very much. I think now my colleague can present.
Professor Vinod, how much time is left for me? I think you are muted.
Sir, I think 10 minutes.
Yeah, you have another 10, 12 minutes, Naresh. Yeah.
Okay, thank you. Thank you for having us in this symposium, and also for organization of a symposium of this kind, where from different disciplines we are having presentations of the important works which have been recognized by award of the Nobel Prize. Can I be heard?
Yes, sir.
Am I audible?
Yes, Naresh.
Okay. So, can I present now? Vijay, can you stop your presentation? So I will try to do it very quickly. I will try to make it shorter. I will be basically having a PDF file with some notes on this, and I will be presenting it now. I hope it is visible. So as already mentioned by my dear colleague, Dr. G. Vijay, that this year's Nobel Prize, or what is more correctly called the Riksbank Prize in Economic Sciences in memory of Alfred Nobel, half of the prize goes to David Card of University of California, and other half, this is for empirical contribution to labor economics. That is what citation says. And the other half goes jointly to Joshua D. Angrist of MIT and Guido Imbens of Stanford University for their methodological contributions to the analysis of causal relations. Now, this is a small illustrative list of some of the contributions, and I will be focusing a little bit on the first of these, the methodology for natural experiments. In particular, in methodology, incorporating instrumental variables from economics and potential outcome approaches from statistical science together. And they also give the concept of local average treatment effect, or what they call LAT date. I'll be spending a few minutes on that, but quickly to the wide variety of contributions they have made, both independently and some of them jointly as well. So causality and policy evolution, their contributions to that, labor economics, many illustrations have been very nicely presented by Dr. Vijay, particularly in context of contributions of David Card. They are related to immigration, schooling or education on the later earnings, the impact of employment protection, how to study this, fertility question and the labor supply of parents, and very, very many other studies of similar kind of thing. On education, there are very nice works. In particular, one I have mentioned is elite impact illusion, that with the peer recognition, what does it say about the quality of education, etc. And they show that there is actually not very significant to their methodology that we're talking about. Voucher system for private schooling and its long-term effect. Seat allocation in education institutions, particularly those where there is a lot of demand. And some of the methodologies are very common, and we'll mention some of them. There are some interesting studies about whether the impact of being war veteran on lifetime earnings. These are some of the early studies they talked about. And very interestingly, they have also contributed to teaching of econometrics, some novel approaches, is particularly starting with the applications and then going on to theories and incorporating, in fact, the natural experiment approaches in their textbooks that they have written, both on econometrics and statistics. Now, causality and experiment, there is a problem of how to isolate the effect of a single factor. Because when you're studying relation between different social phenomena, so there could be multiplicity of influences. Now, in this context, I very quickly mentioned about the kinds of experiments. I am reminded of thought experiment that people have talked about, and especially where they contribute to theoretical ideas. And I am particularly reminded of thought experiments that Albert Einstein was basically involved in, leading to both special theory of relativity as well as general theory of relativity. He was awarded Nobel Prize in 1921, and now another innovation in the experiment in economics and social science, etc., like natural experiments 100 years later. In economics, there is Nobel Prize for these methodological innovations and their wide-ranging impact on the way we can understand the causal relations in economics and social sciences. Then, of course, laboratory experiments are where there is the greatest control available. But in the real world, there are also what we call field experiments and randomized controlled trials, which were referred to a few minutes ago by Dr. Vijay also. So randomized controlled trials, they give more control in terms of the way one can select the population that is assigned to the treatment group, and those were assigned to the control group. Whereas natural experiments, that kind of facility is not available. How they can be useful is what we're talking about. In particular, the important thing is that co-movements or correlation between two variables need not be taken as causation. That is a standard thing that is talked about, but how to get over that. So there, as I mentioned, there is a problem of confounding. Confounding, in a few minutes, I will also talk about in a particular case that how the two variables which are showing correlation, co-movement, they may both be affected by certain characteristic features which impact both the variables in the same way. And hence, it may not be causation between one variable to another, between these two variables, but it may be spurious in that sense. So randomization and RCTs can be an answer wherever it is possible, but for ethical, financial, because RCTs are very expensive to conduct also, or other practical reasons where it's not possible to conduct even if other obstructions are not there. The introduction of this natural experiment methodology has been extremely useful. And this is when it is possible to see variation induced by either nature or policy or institutions. Okay. The variation is of this nature that one can look at the separation of those who are affected by treatment and those not affected by treatment, and as if the assignment of treatment was randomly done. So if this can be ascertained, that without, in fact, implementing any randomization, that it is as if randomization, those are the cases where we can talk about natural experiment or quasi-experiment we talk about. Examples are that, see, for example, even in India, there are studies where you look at the individuals on either side of the state borders, where certain policy that one wants to study, they are different in the two states. So one will assume that they are very similar characteristics, the people who are on the either side of border. And the only distinguishing feature is that policy is taking one value in one state, another value in another state. So impact of that policy variation can be studied. And it is as if randomly some people have been assigned to one side of the border, to state A, and others have been randomly assigned to the other side of the border, say state B. There was an interesting study where low birth weight babies and whether medical intervention is effective. Now, this basically relies on the policy of certain hospitals or state to say that newborn babies which are weighing less than 1,500 grams, they will be given this intervention, and those who are above 1,500 grams will not be. But if you look at those babies which are just below 1,500 grams and just above 1,500 grams, then they should be having very similar characteristic on average. However, it appears that as if they have been randomly assigned to these two categories. And if one looks at the impact of intervention on 1 year mortality rates, this is another example of a natural experiment. Now, for this illustrating this methodology, we take one of the examples here, which is based on Angrist and Krueger's 1990 paper, as well as Impen's and Angrist 1994 paper. This is about years of schooling, that how much causal effect can be discerned on the later earnings of those people who have different level of schooling. And Imbalance and Angrist, this basically provides a methodological grounding later on, and where this local average treatment effect that we talked about has been grounded. Now, years of schooling, we will be seeing that certain features in the law or policy, they are taken into advantage, that how they randomly allocate some people into one group, other group. If we just look at those who are, say, for example, completed high school, and those who have not completed high school, and we look at the average earnings of these two groups, then it can be shown that this will be a biased understanding of the causal effects. And for that, let me quickly, very briefly explain what we are having here. These are symbols which are given. Y is earning, D is education status. This is dichotomous variable, whether one has completed high school or not completed high school. And Y at 1 is essentially for those who have completed high school, earnings of those, and Y0 is those who have not completed high school. I is for ith individual. Now, quite clearly for any one individual, either we will be observing either that individual have completed high school or not completed high school. So both these values will not be observed for any one individual. So if the individual has completed high school, then what that individual would have earned if he has not completed high school is a counterfactual. And this natural experiment is basically taking counterfactuals and how they can be incorporated into the picture. Now, this is where the confounding occurs, that there could be characteristics of individuals which help an individual or motivate an individual to come. Characteristics may also be providing abilities to that individual to have a higher earning abilities. So even if that individual has not completed high school, might have been earning more than some other individuals who did not have the same kind of abilities and did not complete high school. So that is one confounding issue that comes into the picture. And supposing that those who complete high school voluntarily, they also have somewhat higher abilities to earn in the later life, how the biased estimation of the cause can be resulting out of that is first demonstrated here. Suppose we observe that this is what we want to observe. We want to look at the causal effect that any one individual, if one completes high school versus does not complete high school, how much extra earning on average that individual will be getting. So this is to be averaged over all the individuals in the study. Now, the thing is that those who have completed high school, they cannot be the same individuals who have not completed high school. So this cannot be estimated. This is not possible to estimate, but this is what one wants to know. So what actually happens if we look at the earnings of those who have actually completed high school and those who have not completed high school, this is the observed difference that one looks at. Right. So the observed difference is between those DIAs, one means that they have completed high school, looking only at those individuals, that what are their earnings on average, and those who have not completed high school, what are their earnings, okay, if they had not completed high school, as is the case. So if this is observed differences in average earnings of these two groups are looked at, it is shown that why they will be showing an upward bias. That is because that uh, if we look at this figure and we add that if those who have actually completed high school, what would have been their average earning capacity in case they had not completed high school. This is one average that we add or subtract without actually calculating, but as variable we add and subtract. So this remains same as the original observed difference, right? And if we rearrange these four terms, then we get the first, we can get the difference where a person who is actually completed high school, okay, if you look at that person, then what he would have earned or she would have earned upon completing high school and would have earned or not earned if the same person had not completed high school. This is the causal effect actually you want to study of those who are in this particular part of the population. However, the actual observation also at this part, which simply says that those people who have completed high school, what they would have earned if they had not completed, and those who have not completed high school, on average, what they would have earned if they had not completed high school. So if their average abilities of those who complete high school of earning is greater, then this will be positive. And this adds a selection bias. So observed based on the data alone, we will not be actually studying the causal effect properly, and this will be giving overestimates of causal effect of schooling on earnings.
Naresh, do you need some more time or you're almost?
Yes, 3 minutes.
Yeah, if you can just wrap up, please. Yeah.
Right, very quickly, I'll try to wrap up. So what they take advantage of here is that they go for the instrumental variables and they choose those variables which will help us in actually trying to estimate the causal effect that we want to do. And in this particular study, what they do is that entry of the child at the school entry age should be 5 year plus. So it must be in the 6th year of schooling. And there is a compulsory schooling till a given age, say 18 years. So for the entire, and of course, admissions take place at a particular point of time, say in September. So if you take all the people who have completed, say 1960, okay, who have completed, who have been born anytime in 1960 from January 1st to 31st December, then they will be admitted in 1966 September, and all of them. However, some of them will be completing their 18 years without completing high school, and some of them will be completing their 18 years after completing high school, depending on when they are born early or they are born late. So those who are born early, they complete their age 18 earlier, and they can opt out of schooling because compulsory schooling is no more valid. So what they do is that they divide the entire population into those who are going to school in a particular year. Those who are born in the second half of the year, that means they are the early entry, they are at a younger age they enter, giving that value 1 for that variable. And those who are born in the first half, so they are late entry, they are a higher age at the entry. So they divide into these two groups. And this is the instrumental variable, which will determine basically their probability of completing high school of these two groups will be different. Those who are completing, say, they're, they are entering the second half of the year, they will be completing their 18 years of age much later. So they will end up completing high school by law. So there is some people, now one assumption there is that proportion of the people who will want to drop out of school as soon as they are allowed to is same irrespective of when you are born, because your being born is randomly determined by the nature. So that randomness in the nature is taken advantage of. And from this it follows that in the first group, Zi equal to 1, there will be larger proportion of people who will complete high school compared to those who have been born earlier. Now, this, as I said, is this instrumental variable influences your whether you have completed high school or not, and that influences income. Now, in their paper, Imbens and Engris, they provide minimum conditions. The four are mentioned here. That random assignment is one assumption. That distribution of the earnings is independent of the nature of the distribution of this instrumental variable. It should be relevance that ZI should actually impact whether you complete high school or not. Okay, so this instrumental variable is significant. And exclusion that the earnings, it depends on ZI, okay, in such a fashion that whether you are in which year you are born does not matter. Whether you completed high school or not, that is the only thing that matters. Okay, so otherwise it should be same. And monotonicity, that those who have in fact born in the second half, their probability of this is the monotonicity condition. Their probability of completing high school is greater than those who do not. With these assumptions, they show that what they call intention to treat in the context of what they say about the imperfect treatment, that some people who are assigned to treatment group actually may not take treatment. That is called intention to treat effect. That that can be shown to be equal to expected value of incomes when a person is born in the second half of the year compared to the person who is born in the first half. And this can be shown to be equal to that look at those individuals who complete their high school only when they are required, say by the rules or by law. That means that if they are born in the first half, they will not complete, but they will be completing that. Which means there are some people who in any case will be leaving school, they will be dropping out of the school, provided they are allowed to, and only will complete when they are not allowed to. And there are others who will be completing high school in any case, whether there is any legal binding or not. So if you look at those individuals alone, then difference in their earnings gives the causal effect of the schooling or being high school or not being high school on the later earnings. And this is one particular illustration of that, how the naturally occurring randomization in terms of when you are born can be taken advantage of. So we are not looking directly at those who have completed high school, what are their incomes, and those who have not completed high school, what are their incomes. But those who are born in the first half versus those who are born in the second half of the year, we are looking at them. And in the those who are born in second half, the proportion of the people who will be completing high school by the construction that we have said, this will be higher. Okay, probability of completing high school will be higher.
Naresh, we are almost out of time now.
Last line I'm saying, and then I will be concluding. And so that is the only variable we use, and which has a randomization occurring in the niche. This kind of technique has been used in very many applications. If you permit me to say a couple of sentences, that my first paper that I worked on with my supervisors of 40 years ago, nearly, Professor V.K. Chetty. In fact, we were wondering of that kind of thing only, and in fact, that was paper on sugar industry, price and quantity controls in the Indian sugar industry, presented at the 4th World Congress of Econometric Society, where we try to look at that frequent policy changes, 5 policy changes in 15 months in the sugar industry, starting from the 15th August 1978, should provide an opportunity to look at the causal effect in the sense mentioned, but these techniques were not there. We did not develop either. I'm not suggesting that we made any step towards it. And this perception has been there, but there is a landmark contribution. They have developed methodologies that how it can be actually implemented and to study a wide variety of social and economic phenomena. Thank you very much.
Thank you, Professor Naresh Sharma and Dr. Vijay for sharing with us the important contributions of this year's winners of the Nobel Prize in Economics. Yeah, we have gone beyond our time, but if there are 1 or 2 questions, we can still take them before we move to the next prize talk. While we wait, one very quick question to either one of you, Naresh. If I remember correctly, the 2019 prize to Abhijit Banerjee and others was also for their sort of methodological innovations with RCTs in areas like poverty alleviation and so on. So I'm just wondering about the trend in economics prizes that increasingly they are going for methodological work. So is it something to do with the evolution of the discipline itself that there are new innovations just emerging and they are being recognized for the prize?
Actually, methodological.
Either one of you.
Yeah. Methodological innovations have been recognized earlier also, but you are very right that 2 years ago, there was also for the experimental approach to the problems. And there it was essentially, see, this randomized control trials are not new. They have been there for decades, in fact, maybe close to 100 years. The earliest that R.A. Fisher work about the agriculture. In fact, the many of terminology comes from that in the design of experiments field. This treatment is about various kinds of chemicals, for example, and not using chemicals, etc. And how to bring about that effect of the, say, naturally varying fertility of the soil, how that can be avoided. So various designs and randomization were brought about, and that is where treatment and control groups came. And in social sciences at field level, this kind of control and treatment has been used, but through survey methods. Now, what is randomized control trial says that you focus on certain factors where everything else is kept constant. So you actually select and you implement treatment. The other social surveys where you are not implementing, you are studying already existing data. This was one definite, basically to understand the causal effects. See, when you are doing econometrics regressions, this is a big problem, whether it is incidental, which way it is caused. Effectively, we are only looking at 2 variables. Is it correlation?
Thanks.
And there have been time series techniques which they try to do, but they actually try to isolate factors to that. And this is further innovation that when it is not possible. For example, you cannot say that some people should be given a particular kind of schooling, others should not be given. You cannot implement that kind of treatment. Ethical issues are there.
Yes. Dr. Vijay, anything?
Experiments. So that has been basically, but in otherwise also, say in econometrics, methodological schools have been recognized. In game theory, they have been recognized also. Okay. One last sentence I'll say is that it also the kind of problem they have applied these techniques, these particular economists. So they have been very socially relevant problems, whether it is Avijit Banerjee and the two others, the Flow and Framers, or these three that you're talking about. Just look at the nature of problems. They are very, very pertinent today. For example, today, if we have to look at these three farm laws that we're talking about, can we study through some kind of thing?
Dr. Vijay,
they are applicable in the state, they are not applicable. How they impact there?
Anything very quick to add to what Professor Sharma said before we move to the next talk? I mean,
yeah, I think we have already exceeded the time, but quickly, just the idea that in social sciences, unlike in natural sciences, knowledge doesn't really evolve linearly. And what we see in David Card's kind of contribution is what I can call as the Romer movement. With reference to how Keynes was challenged, say around the 60s. And empirically, therefore, we are trying to find the kind of anomalies that exist in the theoretical models in order to then try and improve our understanding, because our theory, in a sense, has for long been stagnated. And we are trying to see if it can be recast, and perhaps that will influence both the policy as well as the classroom teaching. Thank you.
Okay. Thanks, Vijay. Yeah. Thank you both for your contributions to this Nobel Prize in Economics. With the permission of the organizers, let's quickly move on to next prize, which is a Nobel Prize in Literature, which also creates much public excitement. Usually, every year it is announced. This year, the prize went to the Tanzanian novelist, more specific, of Zanzibar origin, Abdul Razak Gurna, and it created quite a bit of excitement for a variety of reasons. No Black African writer won the prize since Wole Soyinka in 1986, and probably no Black writer won it since Toni Morrison in 1993. So for very good reasons, this prize got some excitement. So let me quickly introduce the two speakers who are going to talk about Mr. Gurna's contributions. We have Professor Anna Kurian, who is currently the head of the Department of English. She's a Shakespeare specialist, does work on Shakespeare studies, has published a lot of things, especially for school textbooks through Cambridge University Press. She's also author of a Orient Black Swan book on Shakespeare and has many other contributions in academic journals and books. Welcome, Anna, and joining her is Professor Pramod Nair, who is former head of the Department of English, and Pramod has been hugely prolific. If you, I really don't have time to, recite all of Pramod's publication achievements. There are books. We keep saying that if we don't meet Pramod, for a couple of months, there is a new book. So Pramod has been one of our very prolific humanities scholars. In fact, the only literary studies professor from India to be in the top 100 most cited scholars globally at Stanford University released recently. Pramod is also the director of our Institution of Eminence program at University of Hyderabad. A few years ago, he won the Visitors Award given by the President of India for contributions to research in arts, humanities, and social sciences. So welcome, Pramod. Over to the two of you to use the time as best you think you can to talk about Gurna. Yeah, over to you.
Thank you. Thank you. We'll begin where things usually end, with a vote of thanks, actually. Thank you to the Vice Chancellor for proposing this series. Thanks to the indefatigable Dr. Bindu and Dr. Anil for putting this together. Thanks to Professor Vinod Paural, old friend of the Department of English, if we may presume to say so. Has always been very encouraging, and to our colleagues and friends, and of course to Anna, the usual suspect in most of these ventures of ours. So thanks to all of you. So yeah, as Professor Pravala mentioned, there has been a paucity of Nobels to African authors and African American authors as well. So it was a bit of a surprise and a welcome one when Garna wins it for 2021. It's important to note that it occurs at a particular time in history, which we'll come to a little later. The Department of English has a particular connection to Garna as well. Garna was supervised by Professor C.L. Ines, who is a very good friend of the department, has been a visitor very often. Garna did his doctoral work with her. And Garna's PhD student was a member of the Department of English faculty, Syed Mujibuddin. So there's a lineage of sorts with the Department of English. As a Zanzibar exile, Garna begins his career as a hospital orderly in England, and then goes on to do his studies at the University of Kent at Canterbury and takes his doctorate there. It's interesting and significant to note that Garna is not a literary bestseller. He is known as a very serious writer of serious literary fiction, and is not comparable to the controversial Rushdie, the best-selling Adiga, for various reasons. We will not go into that. And he has a niche audience. He writes a very different kind of post-colonial fiction, not the usual style that you see associated with either Rushdie or with, say, Gabriel Garcia Marquez. So the Zanzibar exile, who begins life as a working class immigrant in England, goes on to become a teacher. He finished his doctorate and was a member of the faculty at the University of Kent at Canterbury, from where he retired as a professor of English. Over to Anna now.
Yeah. So I hope I'm audible. I will begin with the citation because we are focusing on what the citation says about Garna and how that is reflected in his work. So as we all know, the citation says, for his uncompromising and compassionate penetration of the effects of colonialism and the fate of the refugee in the gulf between cultures and continents. I believe that in the 20 minutes or 25 minutes that we have, we will be able to cover quite a bit of what is being said of his work by the Nobel Prize Committee. Now, Abdul Razak Garna has only 10 works of fiction. It's a rather small body of work, you might say. Also because many of those books have fewer than 300 pages. They are like between 200 and 300 pages in length. So when we look at them, and I will briefly go through the 10 titles that he has. So Memory of Departure, his first novel, which is in the late 80s. And then in the 90s, there is this sudden large number in terms of Garna's work. So there are like, I think, 3 books in the 90s. And then these include Pilgrim's Way, Doughty, Paradise. And Paradise was his Booker Prize winning novel. So after Paradise, then there is Admiring Silence, By the Sea, Desertion, The Last Gift, Gravel Heart, and as recently as last year, Afterlives. Also, I think his most sprawling work, about which Pramod will speak in a minute or so. But before that, I would like to point to the one way in which we are approaching the work of Garna, his body of work. We are going to be looking at it in terms of history and geography. Garna, in his fiction, moves from Africa to Europe, and he moves in historical time from pre-colonial through the colonial to the post-colony. And then he shifts us from the post-colony to the experience of the immigrant and the refugee in England. So in terms of both time as well as space, Gurna covers a lot. And some of that is what we hope to touch upon very briefly in the time that has been given to us. Pramod?
Yes. As Anna said, it's a wide temporal frame that Gurna is interested in. He looks at colonial Africa, colonial Europe from roughly the 1880s all the way to the independence movement, post-independent, post-colony in African nations, and of course the immigrant experience. So we can think in terms of three principal themes: colonial history, the nature of the post-colony as in post-independence Africa, immigration, and refugeedom, which involves, of course, studies of Black lives in Britain in novels like Doughty. Pretty much all his novels have something to do with growing up in some African nation or the other. In many cases, they are unspecified, but we will come to that in a while. But his most sustained exploration of colonialism is in a novel called Afterlives, which is his latest text. It's set somewhere in East Africa during the Abushiri revolt of the 1880s, and is fascinating for the way he demonstrates the effects of colonialism, where he says, for example, the Germans and the British and the French and the Belgians and the Portuguese and the Italians and whoever else had their Congress and drawn their maps and signed their treaties. So he's talking about colonialism, the scramble for Africa, as it used to be called, where Africa had been divided up neatly by various companies and their governments from European nations. And what was Africa was essentially constructed in the form of colonies, little bits and pieces engineered by the Europeans. But what is interesting is also the kinds of things that Gurna says about places like Zanzibar. For instance, he addresses the problematic domain of collaboration of the natives with the colonial masters. And in novels like Afterlives, he will speak about people like Hamza, who joined the army, worked with the Germans or worked with the French or the British, and were effectively part of the colonizing apparatus. So Gurna is not a very ardent post-colonial in the sense all blame attributes to the Europeans and the native Africans were wonderful people. No, he doesn't do that. He shows the dirt, the muck that is the empire, but he also points to the dirt and muck which is part of African history, native to it. And that is an interesting point. The second aspect of colonial history that Gurna points to is, of course, the racism in the colonial setup, where when talking about the army, and this is something that contemporary writers have been talking about, like Gajendra Singh and Raghu Karnad, when they speak about Indian soldiers in the British Army and the racism which was used to target them, even though they served the British Imperial forces and served in British Imperial Army. The racism is very clear. The soldiers who come from Africa and join the army are left to fend for themselves, even within the army, while the European soldiers have a relatively easier life. So there is this little component to this. A third aspect is that despite his critique of colonialism, he also notes their evangelical and civilizational mission activities. It's not that it's an outright praise of what they did, but because he refers to them in one of his novels as the gifted killer race, the gifted killer race. And in this, he's castigating the entire white race. But what he also points to is things that they actually did. So in one of his novels, there is a reference to the German missionary compound that is in Afterlives, where the missionary serves everybody who comes up there injured. And he notes that those terrible epidemics that used to occur much more often before the Europeans came with their medicines and their hygiene. And he says every disease you could name was to be found in Zanzibar, including sin and disappointment. So when talking about colonial history, he is not absolving or glorifying the natives, He's not saying that everything was nice before the Europeans came and the European racism is what destroyed it. So he's very conscious of that. My fourth point has to do with a different angle to colonialism that Garna is interested in, and that has to do with non-European support of the colonial project. And in this, in several novels, he will mention the Indians who settled in Africa and enabled European colonialism. For example, in Afterlives, Khalifa's family comes from Gujarat, and Khalifa is a part of the East African business empire. Later, he will say that when the Indians came, and I quote, they are taking over all the commerce and telling the government that they are British citizens and they should not be treated as the natives themselves. And this criticism is constant, where at every point he is emphasizing the fact that the Indians were part of an imperial setup in the East African nations and consequently enabled colonialism to progress. And in this particular process, and that's my concluding point before I hand it back to Anna, the Africans are treated as outside the pale by the Europeans, but also by the Indians who support the British for business interests. Over to Anna now.
Thank you, Pramod. So moving on from the nature of colonialism and how Garna portrays it, we would like to look briefly at the nature of the country after the colonizers went back to their home countries and the post-colony, as we call it. Now, the post-colony figures in the novels that I read, it figures most strongly in Admiring Silence and By the Sea. Garna, and this is where I think the Nobel citation locates it when it says it's uncompromising, his portrayal. He is uncompromising in presenting to us a version of the post-colony which does not, one, attribute all blame to the British and say that we are this way because the whites did this to us. Two, it demonstrates how there is friction among the varying people groups who are located in the post-colony. And in Garna's books, because they are set in Zanzibar, you have not just the Africans, and he says extremely rude things about the Africans who come from the main continent. He also says, he speaks rudely, shall we say, about the Arabs, the Indians, of course, and he calls them Bania. So all the small people groups which together people Zanzibar, he brings to us a reflection of their lives after the departure of the British. And in that portrayal, he then shows us how the country sinks into utter chaos and anarchy. A chaos and anarchy, which he also makes clear is not because the British gifted it to the people, but because the people themselves are so torn apart that there can be nothing other than chaos and anarchy. Much of the blame, of course, he lays at the door of the ruling classes, whom he castigates as being vengeful, violent, indulging in their petty revenge sagas with people who sneered at them once, who looked askance at them once. So he then proceeds to show how the post-colony becomes this mire, and in some cases, in some of the books, especially in Admiring Silence, if I remember right, it is actually miry, filthy, because he speaks a lot about blocked toilets, blocked plumbing which doesn't work, and how the whole country is a stinking cesspool because everything, beginning from the plumbing but going up to government, has broken down. The interesting part, of course, also is that the post-colony's nature does not change. So in the years immediately after the colonizers leave, it is this way. So that's in a book such as By the Sea, you see it immediately after the colonizers leave, but you also see it 20 to 30 to 40 years after. So in Admiring Silence, which is in the 1990s, he goes back, the main character, the unnamed narrator of Admiring Silence, goes back to his country. And then he discovers that it still remains the same. There is still vengeance and violence. There is still rape, murder, torture, anarchy. There's still no roads, There is no progress. Cooling is still insufficient. At that point is the point, I think, at which the citation says it is uncompromising, because he does not provide the usual glossy picture of the post-colony, or even if not glossy, the progressive picture of the post-colony. Instead, he shows us the breakdown of the post-colony. Pramod, would you like to add something?
Yes. One is that in the post-colony, he has several jibes to make about the so-called nationalisms that arise. And at one point, in as early as his first novel, Memory of Departure, when Omar the hero meets Moses on the train, Moses is ranting and raving, and he says, all we have is African art, African literature, African history, African culture. And he says, and we can't even make a screwdriver or a tin of talcum powder for ourselves. Everything we use, we get from Europe or America. And then we talk about tradition and African this and African that. It's a take on the intra-tribal tensions that continue to haunt the post-colony. It's not all just the Europeans left and things are fine, but the older rivalries have returned. And the point is that the duplicity that marks all this. During the colonial period, the Arabs and the Indians own all the land and all the businesses, he says, in afterlives. And the blacks are skivvies and laborers. Then finally he will say, oh, as for the democracies in Africa, in one particular novel, Afterlives, he says, they were about socialist one-party democracies, he says, they were an abomination. And this is one of the most sustained comments Garna will make about independent South African nations. He calls them an abomination, an anathema, thinly disguised dictatorships. And then people say, oh, but they are our people and for national unity. And that is when he writes this, and I quote, for national unity, he read national bullying. Their enthusiasts described them as traditional forms of government. And he asked, whose traditions? If some greasy king had managed to gain ascendancy over his own people and molested and harassed them for some bizarre ambition he harbored, was that a reason to follow in his bloody footsteps? All of this has to do with how the post-colony is not really free of the intra-racial, intra-tribal, intra-community tensions. And it has nothing to do with the Europeans. So the blame, I'm getting a lot of crosstalk. Somebody's mic is on.
Can we please mute the others? Yeah, go ahead.
Thank you. So the post-colony is rife with the exact same violent genocidal acts as it used to be before the Europeans came. And once the Europeans leave, the new rulers take over the country and run it exactly that they were used to, which means the genocide, the violence, and the rape repeat. And nothing is quite all right in the post-colony. As for nationalism, less said the better, which is what Anna was referring to as uncompromising. It is not, and it reminds you strongly of a very early novel, 1968, Arma's novel, The Beautiful Ones Are Not Yet Born, in which the disillusionment with independence is already in, a little bit like the Hindi poet Pradeep's poems. 10 years after independence, he was already mourning the rise of a new India and what it represented. Back to Anna now.
Professor Pavrala, we have about 10 minutes or 15?
Yeah, if you can take another 5 minutes, Anna, to...
5? Both of us together, 5?
Yeah, I mean, if you need a little more, that's fine. But then, you know, we have some time.
No. We'll try and do this within 5. We'll wrap it up. So we would like to move on then to the experience of the refugee and the immigrant. And that both of us believe is the reason why Garna also gets the Nobel at this point in time. One, of course, because he's speaking of the Black experience, but he's also speaking about the refugee experience. And given the last few years, actually not just few, but given our sustained questioning of the status of the refugee, which we saw as recently as Afghanistan and Poland, but also over the last few years, the influx of the refugees into Europe. Garna focuses on the beginnings of that time. He focuses on the 1960s and 70s when people came into England and how the English felt themselves being swamped by the refugees. But at the same time, when he speaks about the refugee experience and the immigrant experience, he does not make it a story which is always in line with the positive side for the refugee and everything else being negative. In fact, he shows us the refugee struggling. He shows us the refugee stereotyping the whites. And he shows us the empowering of the refugee when he or she, and usually it's he in Garna's fiction, when he struggles but eventually manages to make a small safe space for himself in English society. That small space, however, is made at a considerable cost. The refugee and the immigrant in Garna's fiction is usually neither here nor there. He is in this kind of liminal space wherein he stands at the doorway to both cultures, is fully participatory in neither, and finds himself an exile from his home and his country, whether that is England or whether that is Zanzibar. And this again can be seen in books such as By the Sea, Admiring Silence, The Last Gift, even Dottie, where the immigrant and the refugee, they come into the country, they hope to make a place for themselves. They sometimes succeed, but those lives are small scale, melancholic, bleak, on the whole, not characterized by these grand success stories, which otherwise we like to talk about. We love to say that the refugee made it big. The refugee in Garna does not make it big. He is content with a very minuscule life. That minuscule life is also fraught with sorrow, with the glossing over of ancient histories and stories that he once had, and is instead located in the here and now. Pramod, I think, will now sum up and we'll be done.
Yeah.
Pramod?
Yeah, thank you. So he refers to a refugee in By the Sea as the half-life of a stranger. And like Anna said, they don't quite fit in, but in some cases, he also shows a certain amount of cunningness in the immigrant, where in By the Sea, the immigrant, the refugee arrives there, pretends to not know any English. And when finally he's asked, he says, oh, but I was told I should not admit to knowing English. Otherwise, you would not give me a refugee status. And from there to acculturation, small lives. The point is also that there is a reverse stereotyping. They, Daud in Afterlives stereotypes the British and doesn't mind taking help from them. There are British friends who leave food for him and bring him gifts. He takes all of them and then abuses them. He's, of course, troubled by skinheads and racist groups. But all in all, the ability to portray small lives, what the other major novelist, Graham Swift, said, the hidden poetry of small men's lives is what Garna is interested in. It's hidden poetry, it's small lives. And he does not rise to grand heights about hybridity, acculturation. He's not interested. The discomfiture of trying to fit in, but also, and this is what is deeply troubling, of having been an immigrant in Africa is itself a bit of a bother, because those who have immigrated within Africa are themselves treated badly, not necessarily those who migrate to England. So the people in East Africa harassed by the Indian settlers there, by other Africans, and of course the Arabs. So it's a far more complex scenario that he represents. My last point is, in his later novels, especially By the Sea, he's also conscious of American neocolonialisms and what they do. And in this, of course, to reiterate the point I made, there's a rejection of the so-called nationalisms of these post-colonial states, where he says, we don't even manufacture talcum powder and we call ourselves nationalists. We don't make anything that works. Not one thing works. And this has nothing to do with Europe. But he's also afraid of the fact that the CIA operates in many of these territories. There is a kind of uncertain consorting with and cohabitation with local regimes, which enable further exploitation. So in Garna's portrayal of the immigrant experience, there is never grand dreams. There is just survival. And survival is the hardest thing to do. We'll close here, so we'll have time for questions. Professor Bavarala, you're on mute.
Yeah, thanks, Pramod and Anna. Thank you for throwing light on the work of Abdul Razak Garna, which won him the Nobel Prize this year. As it so happens, some of the works of Nobel laureates are usually not widely read by the general population until the prize is announced, and there's now a lot of excitement and people want to get hold of their books and so on. So I'm sure some of that is bound to happen with his work. But one of the things we could talk about, I mean, there could be others who may have questions, but the fact that only, apart from Garna, there are only 5 other Africans who were given Nobel Prizes before for literature, it is something quite interesting, and it could lead us to some discussion about the politics of, literary prizes. I mean, you think about Gugi Wathiango not getting a Nobel Prize until now. But someone whose body of work is actually not as voluminous as many other writers makes it. Is it something to do with the themes of someone's work at a particular time, the themes of exile and immigration and the refugee lives and so on, appeals to the jury at a given time, or I don't know what goes on in the decision making about the literature prizes in particular. I don't know. Just a comment, yeah.
Yeah, we actually had a segment on the context within which we can read Garna, and the 2 contexts which we had was one, of course, the problem / the question of the refugee, and the other one was the Black Lives Matter movement. And it is against those contexts that Garna wins his prize. But another, I mean, tangentially saying the same thing, is to think about how you have Booker Prize winners at a certain point, always a certain kind of person. So Margaret Atwood winning it last year with her reprisal of The Handmaid's Tale, a sequel to The Handmaid's Tale would be one example, but winning it alongside Bernardine Evaristo would be an example. That there is a point at which a certain kind of book or a certain kind of author becomes a viable contender for the prize. Pramod will have something more to add to this, I'm sure.
Well, yes, the politics of prizes, as Professor Bavarala put it, it's a huge area of study. There are books written on the Booker books. And of course, the Booker's hoary tradition of being a colonial enterprise, running a sugar company decades ago. That apart, the contexts are crucial. There has always been some puzzle as to why, for example, Naipaul gets it at a particular point in time, or Derek Walcott, who is Caribbean British, and you discover that there are problems with how these prizes get allotted, some to East European novelists and poets who are, as you yourself said, sir, not very widely read. They are not available in many cases in translation. In fact, in the case of Garna, there was a report which said people were rushing to the bookshops to find his books and they couldn't find it because they are not best-selling works. And you see Rushdie winning Bookers, then the Booker of Bookers. The Swedish Academy's method of awarding prizes is a closed secret. The final announcements and the people who were involved can be released only 50 years after the award. So you never get to know. But definitely the context that Anna mentioned have a role. He speaks to the moment.
Yeah.
And in the last 5 years, other than climate change, and we expect that there will be at least one novelist writing cli-fi, that is climate change fiction, will be getting a Nobel soon, because in the wake of the IPCC, that's the next big thing after refugees. So there is something to... Ghosh is a likely contender. He's got the Gyanpith, but he's a likely contender, especially because of his non-fiction and his new work, The Nutmeg Scars, also.
Yeah. Okay, great. Thank you. I think Professor Ramanamurthy had raised his hand.
Yeah. Am I audible?
Yeah.
Yeah. Congratulations, Pramod and Anna. I liked your presentation, and I'm provoked to ask a simple question that I'm inspired to read this author, and I will definitely try to read. But from your interpretation, I got a sense that it's nice to find an African author who's trying to be self-critical about their own nationalism or sense of nationalism, African nationalism. But there seems to be some gross underestimation of the power of colonialism of advanced nations on countries which are at a very early stage of historical development. It looks there seems to be some kind of underappreciation of that power equation in his understanding from your interpretation. I mean, that's something.
No. Actually, what he does is to try a little bit of balance, which, as you have pointed out, is not very easy to achieve, because the pernicious effects of colonialism, whether it is language, literature, the social order, has been well studied. What Garna refuses to do is to glorify something like a pure African society, which was wonderful. And as he represents it, it is not entirely the doing of the Europeans that has made African society what it is. Some of these flaws are, so to speak, homegrown. So it is a very difficult balance, to borrow a Philip Larkin phrase, that he's trying to achieve, that you can't say all of the faults lie with the colonials. Of course, they are terrible, and the pernicious effects of the post-colonies influenced by, say, the American neo-colonial movement is equally bad. But it's not like these are wonderful societies in and of themselves. Yeah.
We have one question, probably the last one from Professor Pathak, and then we'll move to the next session. Yeah, please.
Mine is not a question, but just an appreciation from a person who has no knowledge about this thing, but also a first-hand experience about this, what was mentioned about the Indians doing in Africa. First-hand experience because I was in England in 72 to 74. And some of the Gujarati Indians who were driven out by the Amin from Uganda, by talking to them, I could see that what Garna has written is perfectly okay, because Indians were thinking themselves as part of the British. And these people who were driven out of Uganda, they were talking like that. So I can see, I am not an expert, and thank you for updating a non-expert in this, Pramod and Anna.
Thank you.
Yeah, other thing is that, well, I was a little disappointed in the beginning of the seminar when I could not join at 10 o'clock. But that is, I think, when I tried to join, because I wanted to understand the physics and chemistry Nobel Prizes, but I could not join. Because when I joined, it said too many people were already there. So I think in future, we have to take care of that. Thank you, Vinod, and thank you.
Thank you, Professor Pathak, for joining us. Yeah.
Thank you.
Thanks, Pramod and Anna, for that fascinating presentation, and at least arousing the interest, hopefully, of the audiences in the work of Garna. And thanks a lot. Yeah?
Thank you.
Thank you very much.
Yeah. Let's move to the last prize category under discussion today, last but not the least, the Nobel Prize, Nobel Peace Prize. It's probably the most anticipated, most eagerly awaited prize every year. People start speculating, and then there's a lot of interest in who wins the Nobel Peace Prize. Of course, people like Mother Teresa won it in 1979, the Dalai Lama in 1989, and Kailash Satyarthi in 2014. And very famously, the one Nobel Prize that was never given to an Indian was Mahatma Gandhi. And there is still writing and speculation about why Gandhi was not given that prize. And this year, the prize went to two journalists. It was a complete surprise. And maybe in some ways not a surprise. Maybe Dr. Rajesh, who is going to talk about it, can tell us about it. One interesting fact about the Nobel Peace Prize, it's administered by Norway and the Norwegian Parliament, unlike all the other Nobel Prizes. So that's another sort of curious fact about Nobel Peace Prize. About Dr. Rajesh, who is going to speak about this year's winners. He's a faculty member of the Department of History at University of Hyderabad. And Dr. Rajesh's work has been on Tibetan history, Tibetan society. His publications have been quite a bit on Buddhism and Tibetan history. And probably quite appropriate for him to talk about the Peace Prize. Over to you, Dr. Rajesh.
Thank you, Professor Vinod. And firstly, I would like to thank the respected Vice Chancellor for having taken the initiative to organize the 2021 Nobel Symposium at the University of Hyderabad. Am I audible, sir?
Yes, you are, Rajesh.
Thank you, sir. Then I'll start sharing my screen. I would also like to extend my heartfelt thanks to all the audience who have been waiting since the morning, that is esteemed professors, dear colleagues, and also the students. And right now I'll start my presentation. Is a presentation, can you see the presentation, sir?
Not yet, Rajesh.
No, okay, sir. 1 second. I'll just, yeah. There's some issue. Okay. It's a small issue. I'll just get it right in 1 second, sir. And there is some issue with the presentation. Yeah. Yeah, now can you see the presentation, sir?
Yes.
Yes. So the
yeah, please go ahead.
Firstly, thank you, Professor Vinod, for your generous introduction. In addition to the Department of History, I'm also a joint faculty in the Center for Human Rights.
Yes, I should have mentioned that. Yeah.
In the School of Social Sciences, we have a PhD program here and also a postgraduate diploma in human rights. This is what spurred my interest to the Nobel Peace Prize. Okay. So this year's Nobel Peace Prize is jointly awarded to Maria Ressa from Philippines and Dmitry Muratov from Russia, both of whom, as Professor Vinod had rightly mentioned, have contributed to journalism, but in a very different way, not unlike the dominant commercial genre of journalism. And this is where they have made their significant departures from the world of journalism, but at the same time, serving the cause of journalism and being glued to the masses. And this is how they have done it. I'll first read the citation, as the Vice Chancellor was very eager to talk about the citation. The announcement of the 2021 Nobel Peace Prize, the official citation goes thus. The Norwegian Nobel Committee has decided to award the Nobel Peace Prize for 2021 to Maria Ressa and Dmitry Muratov for their efforts to safeguard freedom of expression, which is a precondition for democracy and lasting peace. Ms. Ressa and Mr. Muratov are receiving the Peace Prize for their courageous fight for freedom of expression in the Philippines and Russia. At the same time, they are representatives of all journalists who stand up for this ideal in a world in which democracy and freedom of the press face increasingly adverse conditions. Maria Ressa uses freedom of expression to expose abuse of power, use of violence, and growing authoritarianism in a native country, the Philippines. In 2012, she co-founded Rappler, a digital media company for investigative journalism, which she still heads. As a journalist and the Rappler's CEO, Ressa has shown herself to be a fearless defender of freedom of expression. Rappler has focused critical attention on Duterte regime's controversial murderous anti-drug campaign. The number of deaths is so high that the campaign resembles a war waged against the country's own population. Ms. Ressa and Rappler have also documented how social media is being used to spread fake news, harass opponents, and manipulate public discourse. Dmitry Andreevich Muratov has for decades defended freedom of speech in Russia under increasingly challenging conditions. In 1993, he was one of the founders of the independent newspaper Novaya Gazeta. Since 1995, he has been the newspaper's editor-in-chief for a total of 24 years. Novaya Gazeta is the most independent newspaper in Russia today, with a fundamentally critical attitude towards power. The newspaper's fact-based journalism and professional integrity have made it an important source of information on censurable aspects of Russian society, rarely mentioned by other media. Since its startup in 1993, Novaya Gazeta has published critical articles on subjects ranging from corruption, police violence, unlawful arrests, electoral fraud, and troll factories to the use of Russian military forces both within and outside Russia. Novaya Gazeta's opponents have responded with harassment, threats, violence, and murder. Since the newspaper's start, 6 of its journalists have been killed, including Anna Politkovskaya, who wrote revealing articles of the war in Chechnya. Despite the killings and threats, editor-in-chief Muratov has refused to abandon the newspaper's independent policy. He has consistently defended the right of journalists to write anything they want about whatever they want, as long as they comply with the professional and ethical standards of journalism. Free, independent, and fact-based journalism serves to protect against abuse of power, lies, and war propaganda. The Norwegian Nobel Committee is convinced that freedom of expression and freedom of information help to ensure an informed public. These rights are crucial prerequisites for democracy and protect against war and conflict. The award of the Nobel Peace Prize to Maria Ressa and Dmitry Muratov is intended to underscore the importance of protecting and defending these fundamental rights. Without freedom of expression and freedom of the press, it will be difficult to successfully promote fraternity between nations, disarmament, and a better world order to succeed in our time. This year's award of the Nobel Peace Prize is therefore firmly anchored in the provisions of Alfred Nobel's will, Oslo, the 8th of October 2021. The particular statement synoptically captures the whole idea of the need for freedom of expression and the travails faced by both these awardees under adverse conditions. And it talks more about the context, the difficult context in which they have been operating, right from their opponents using raw power to sophisticated power of the internet, like both of them are grappling this whole idea of troll factories. We first talk about Maria Ressa. Maria Angelita Ressa, that's her Tagalog name. She was born in 1963, is a Filipino-American journalist and the author and co-founder and CEO of Rappler, the first Filipino Nobel. She's also the first Filipino Nobel Prize laureate. Maria Ressa was born in the Philippines, but grew up in the US after her family migrated to USA at the age of 10. And there she was exposed to a multicultural environment early in life, like many Filipinos, but with a difference, because Philippines is one of those few countries where the economy relies on remittances from abroad. So you have a very large number of Filipinas working as nurses in West Asia. Then you have many Filipino men working as construction labor in other Southeast Asian countries and in all the service sectors. So it's a kind of cultural hybridity in Philippines, which is a topic for debate. And you also have an academic side of this debate, which is going on in US. So Maria Ressa, while graduating with a BA degree in English and certificates in theater and dance from Princeton University, where she was exposed to the emergent debates in the human sciences, brings up this whole paradox of what we call the different cultural pulls and pushes in Philippines. And these are necessary to understand the context of Philippines. For example, we have the power of the Catholic Church in Philippines, we have the power of NGOs, and then we have what we call the power of a money order economy. That is a large number of remittances coming from outside, and you have people sending these remittances, becoming the role models and justifying that the ends are fine and we should not pay heed to the means. So these were some of the questions which were troubling Maria Ressa, and in contrast to many of these arguments which are made from a very commonsensical platform, she tried to give an academic twist to this while placing Philippines firmly in the global map, that is the global forces which operate in the Filipino, constituting the Filipino experience. Maria returned to Manila on a Fulbright fellowship in 1986 and worked for the government station, that is the television station, People's Television 4, as director of newscasts and then as head of its special projects team. At the same time, she was also associated with a university in the Philippines. Now, the quotation she gives is that after receiving the Nobel Peace Prize, this is really for all journalists around the world. From television to cable television, that is after 1991, we had the first Gulf War, and then the cable television boom took place in many parts of the world, including Philippines, where, as I told you, many of the Filipinos were working in West Asia, and then there was a hunger to know about the situation in West Asia. So cable television became widespread in Philippines. And what really took off was a new media in Philippines, and she witnessed the shifts in these media, mass communications, and smoothly transitioned to carve a niche in the emerging digital media. The digital media is a fort, and then at the same time keeping alive academic interest. One of the peculiarities of Philippines is that it is a country which is composed of many large number of islands, around 13,000. So you do not have direct access of cable TV, etc., to many of these islands. And so the new media is one of the most important elements that gives connectivity to many of these people. This is exploited by most of the companies. Reza was featured on the cover page of Time magazine in 2018 as the Person of the Year, as one of a collection of journalists, that is, a Guardians, as the journalists were called, which is there in the cover, the Guardians and the War on Truth, for combating fake news. So, fake news by this time has made deep inroads in Southeast Asia and also dictated the political ideas there. A journalist in Asia for more than 35 years, Maria Ressa co-founded Rappler. The official name is Rappler.com. This is a digital-only news site. That is, it doesn't have any print edition. It's a digital-only news site, and that is leading the fight for press freedom in the Philippines. Maria Ressa says that in contrast to oral narratives or in contrast to print narratives, the digital platform gives Rappler a freedom that is actually part of the hypertext protocol. That is, people can click anywhere and then they can go to anywhere. So this hyperlinks gives people the way they can craft their narratives of the stories they want to read, rather than it being imposed from above. And this imposition from above is what she's contesting. And has, though Rappler started in Facebook, it went against Facebook after she understood the whole logic of Facebook and the economic situation in which it operates without any need for ethics. Rappler started as a Facebook page and rode on the popularity of the social media craze, because Facebook became the top website in Philippines when Facebook offered free data through mobiles and also through the laptops and PCs through its app. So once you subscribe to a Facebook app, you get free data. And in this process, what happens is that the real motive of Facebook was to entertain advertisements. And this, as we see in the cover page of Time, the War on Truth. So large number of groups, controversial, and some of them with dubious credentials, latched onto this, and many were supporters of the Duterte government, that is Rodrigo Duterte, the strongman of Philippines. He exploited Facebook to the maximum, that is Duterte's aides and many other organizations, including paramilitary organizations, since Philippines also has quite a share of conflict. That is, we have the conflict against the Muslim minority in Mindanao and also against the leftist rebels in some other islands. So what happens is that many of these organizations peddled spam and fake news. So at one point of time, it became so strong that fake news became a dominant idea in Southeast Asia, particularly in Indonesia, Philippines, and Singapore, where you had ideas of tsunamis going to come, and then people started huddling around. This led to a break with Facebook, and she became a trenchant critique of Facebook and a sane voice for the regulation of new media. This critique of Facebook was not only in action by starting Rappler.com, but also she took it to an academic length and then published a monograph, which ultimately became a full-length book. As Rappler's CEO and president, Maria has endured constant political harassment and arrest by the Duterte government and forced to post bail 10 times to stay free. That is, she makes this whole idea that whatever happens online doesn't stay online, which is one of the main arguments she's talking about. Whatever happens on social media doesn't stay on social media. Online violence leads to real world violence. That is, she's referring to what happens that whatever she has written on Rappler.com has repercussions in the real life. The repercussion is the arrest warrants against her and she's forced to take bail. So one of her first books was based on her academic training was Seeds of Terror. This along with the veteran journalist Elizabeth Rubin, who started this particular idea of network warfare. She tries to understand the account of Al-Qaeda spread in Southeast Asia. And then the second book is on From Bin Laden to Facebook. So her whole complaint is against how Facebook, which tried to talk about a very egalitarian digital environment, has in fact become a monster trying to actually leverage people and their micro experiences. So she says that this has gone unheeded. And in 2016, I have felt like, since 2016, I felt like Sisyphus and Cassandra combined, repeatedly warning that a dystopian present is your future. American biologist E.O. Wilson said it best. We are facing Paleolithic emotions, medieval institutions, and godlike technology. She's talking about these two characters from Greek mythology, one who rolls a stone uphill, and then when he reaches a summit, he is forced to do it again. That is Sisyphus. And Cassandra knows what is going to happen, but no one would believe her. So she's saying, I'm doing this job again and again, warning against the authoritarianism of our president, but no one is believing me. So she's caught up in one particular predicament. And why is this message not reaching people? Because social media, with its highly profitable micro targeting, that it has initiate targeting, has become a behavior modification system. And we are Pavlov's dogs experimented on in real time with disastrous consequences. Facebook is the world's largest distributor of news, and yet studies have shown that lies laced with anger and hate spread faster and further than really boring facts. Like the President Duterte, who was addicted to shooting people and confessed that he had shot people ever since he was a student. And he said that the first time he shot someone was because someone had talked to him something bad about his background from the Visayas. He comes from the region known as the Visayas. So this particular idea which was shown in Facebook was actually extolled as an aspiration of masculinity by Duterte. All these were unmasked by Rappler.com. Okay. With press freedom under threat in Philippines, and most of these could not be explained in Rappler.com because they were already having many arrest warrants, a film was made. The title of the film is 1,000 Cuts, and this goes into the escalating war between the government and the press. The documentary follows Maria Ressa, a renowned journalist who has become the top target of President Rodrigo Duterte's crackdown on the news media. So Duterte has become a hero in Philippines to many because of the declining situation, law and order situation in the Philippines, and also Philippines has become one of the most important places for drug consumption. And so Duterte's strong arm methods have won him praise. But the point is that in addition to winning praise, the number of killings and extrajudicial killings has become so high that it has become like a warlike scenario with no one questioning. And so what Duterte is doing is that on the ground, he is killing criminals, or so what he claims. And in social media, this is actually packaged off as trying to restore law and order and make Philippines a better place for the common man. This is targeted to different audiences, and then the public have lapped it. So there are only few voices who are standing out against Duterte. And this is where Maria Ressa has brought a third book, which is going to be published in 2022, How to Stand Up to a Dictator. And now, even after winning the Nobel Prize, Duterte has warned that the Nobel Prize can't be like a shield for her. And in these days of authoritarianism, Duterte has teamed up with many of the dictators around the world. And one of the dictators who is going to be the part of the second story is Vladimir Putin. So here we have this photo of Duterte shaking hands with Vladimir Putin. So Vladimir Putin's nemesis is supposed to be Dmitry Andreevich Muratov, whom Putin hates, and whose magazine newspaper Novaya Gazeta was targeted many times, very violently.
Rajesh, you may have to make this a little briefer.
Yes, sir. It will be only 10 minutes or 5 minutes, sir.
Yeah, please. Thank
Dmitry
you.
Muratov was born in 1961 and was a Russian journalist. Muratov founded the pro-democracy newspaper, and in 1993, this was a tumultuous year because 1991 we have the implosion of the Soviet Union and then the rise of a new Russia. So Muratov studied at the Faculty of Philology and for 5 years, and there he discovered his interest in journalism. And after graduating from the university, he worked in the Soviet Army in 1985. This was a period when you have the troubled years of Soviet Union, the period of perestroika and glasnost. And then Muratov says that in 1993, He and 50 other colleagues from Komsomolskaya Pravda. Komsomol is the organization of the young communists, and this was the day when, after 1991, they couldn't grapple with many of these questions. That is the critique of Stalinism. That is why Muratov was forced to leave it. And with 50 of his colleagues, they started a new newspaper titled Novaya Gazeta. The term Novaya means new. And then Muratov says in his citation that, in his reply to the citation, the peace prize is for my paper, not me. After enduring years of harassment from 1993, he stepped down in 2017 because of threats and the exhausting nature of running the newspaper. Sometimes there would be power cuts and then there would be no gas, and to work in the cold in Russia was very difficult. But after popular appeal from the staff and all the readers, the staff voted for his return in 2019. During his time, 6 journalists have been killed. One of the most important journalists was Anna Politkovskaya. Politkovskaya covered the Chechen War, and the others who were killed were Yuri Shekochikin, who covered corruption, Igor Dominiko, and then Anastasia Baburova, Stanislav Markelov, and Natalia Estemirova. Why Muratov is specific in naming all these people is that he says that the Nobel Prize is awarded only to living people and not to people who are dead. So therefore, it is my duty to mention these people. And Yuri Shekochikin was one of his best friends. The point what Muratov was making, that is, these 6 people are making about is the whole victory in Chechnya, that is after the Second Chechen War in 1999, the linguistic consensus for this war was raised in both the news and also in the new media by Vladimir Putin. Putin came to the power because Yeltsin chose Putin because he was a strong man, and Putin cast himself in the image of the strong man. Now, I'll end very soon, Professor Paurala. Now, the others who died, all of them were killed with either shot outside their apartments or killed with very costly poisons, which is one of the trademarks of the earlier KGB regime and also KGB institution and also the new institution. Like Duterte, who threatened Maria Ressa, saying that the Nobel Peace Prize cannot guarantee her immunity, the same goes with Vladimir Putin. Putin says that if Muratov covers himself with the Nobel Prize like a shield to violate the Russian laws, he does so consciously. Putin said at a public event in Moscow. The other words which Putin used were foreign agent. Foreign agent is one of the most favored terms in Russia to label anyone as an agent of the West. This started during the Cold War and went on. Now, so much of pressure is on Muratov, and we had seen that he had resigned, and only after popular appeal he had to actually resign his resignation. Muratov, Putin said that if he doesn't break Russian law and there's no reason to declare him a foreign agent, this is the whole crux of the matter, then that won't happen. Muratov replied, I am not Pasternak, referring to Boris Pasternak, who won the Nobel Peace Prize in 1958 and was forced to renounce it, that he wouldn't receive the Nobel Peace Prize. Pasternak also did not go to the West because he was so tied to Russia. But Muratov is tied to Russia, but he says that I'm not Pasternak, means I won't be intimidated. So with this, I end the presentation, and thank you all for bearing with me patiently. Thank you, Professor Paurala. I hope I kept the time.
So you're muted.
Sorry. Yeah, I mean, after listening to Dr. Rajesh, it probably becomes more apparent why the Norwegian Nobel Committee decided to award this year's Peace Prize to 2 journalists, Maria Ressa and Dmitry Muratov. I think there's a larger statement that's being made here about the importance of safeguarding freedom of expression as a precondition for democracy and peace globally, and also a statement about the sort of authoritarian populism that is visible in different parts of the world. Rajesh has talked about the context of the Philippines and Russia, but one could also bring in the situation in Turkey, places like Hungary, and so on. So thanks a lot for that. brief presentation on the contributions of these two prize winners. Any questions for us? I know we got into lunchtime and so on. People must be getting restless, but if there are any comments or questions, we can take one or two questions.
Dr. Ramanamurthy.
I think Ramanamurthy's hand probably has been raised from previous time, I think. I'm not too sure if he has a question to Rajesh also. But I see Yogesh Sharma.
Yes, sir. Am I audible?
Yes, yes, please.
So it's a more of a comment rather. I think the Facebook example is a very relevant one in our own context in India. I think probably we know the controversy when Facebook joined with the Geo to offer some free data, and also the Facebook is the biggest minority shareholder in a Reliance conglomerate. So it's a as a aware citizen, we should be watchful of consequences can happen with this kind of media coming together with multinationals like Facebook. Just my comment and observation on this topic.
Thank you.
Yes.
And also, I mean, Maria Resa's work on digital platforms also gives us the potential of citizens being able to use these digital platforms to express themselves more freely. Just not just within mainstream media, but also across these digital platforms. Dr. Rajesh, any response to that comment here?
Yeah, Maria Ressa actually talked about this in her work on the Indo-ASEAN Joint Conference, in which India and China were invited as members. This was the first time China was opening up, and very cleverly what China does is that we see, unlike the whole world, Facebook has become a phenomenon in the English-speaking world. But what happens in China is that we also have other websites like Baidu.com or WeChat that seems to be the mother of all websites in China. It is so interconnected. But skillfully, very few people talk about that. But it has a great spillover effect in Southeast Asia, particularly in Philippines, because we have very large number of Chinese minorities, and these people are seen as a source of trouble and also a safety valve to channelize hatred by the Duterte regime. So they are caught between Facebook and they are caught between WeChat and Baidu. So, yes, thank you so much.
Thanks, Rajesh. And I think we will have to start there now. We've already exceeded the time, but thanks to the organizers and to the panelists in this session for making it a very informative and educative series of talks on this year's Nobel Prizes. So on my behalf and on behalf of the organizers, thank you all for your presentation. Over to Bindu Madhav Reddy and the organizers.
Thank you very much, Professor Vinod Paurala. It's my pleasure to give a vote of thanks for this event. First and foremost, we thank our Vice Chancellor, Professor B.J. Rao, for his vision and plan to have this symposium. And we're extremely delighted to have Professor Paurala and Professor Kumaraswamy to be organizing and hosting this event. We thank them. And we thank the deans of respective schools for nominating the best speakers who aptly represent the areas of the Nobel laureates. And we thank those speakers for delivering exceptional talks and very inspiring talks, especially graduate students who might have listened to these lectures. They jumped in joy with this learning, this kind of discoveries and discussions. And we thank the students of Dr. Bindu and myself, who are helpful for us during different occasions. And we thank all the participants and asking the questions to make this event very vibrant. And at the end, thank you so much for all of you and for the SERP for funding this event. Thank you, sir. Good afternoon.
Thank
Have
you.
a great weekend.
Thank you.